This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 588, recorded on February 21st, 2020. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's sunny, not a cloud in the sky, and it's 32 Fahrenheit, which everyone should be able to convert to Celsius, being 0.
That's the easy one, right?
Yep.
Really good. It's the same here. It's 1 degree Celsius. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing? It is 52 degrees Fahrenheit and sunny here, so nippy by Texas standards, but a beautiful day. I don't know what that is in Celsius. I haven't, converted it. Do it yourself.
All right. That's it for now. Dickson will be around later. If you like what we do here on TWiV, consider supporting us. Microbe.tv/contribute. More about that later. I was just thinking this morning, what we do, you really can't find anywhere else. Where else will you find a bunch of virologists talking for 2 hours, mostly these days about coronavirus, but mostly about other viruses. Plus, you can write in and get your question answered. Mm-hmm. That hasn't happened on NBC or CBS or MSN or Fox or any of that, right? You can't write in. And so check it out, microbe.tv/contribute. Oh, I thought by putting up a non-corona episode, we would hear howls, but there was just one. So
we had no howls?
No, one person on the website.
Oh, one howl.
No mention of coronavirus. Well, it was recorded in October.
It was a good episode. I enjoyed it.
Oh, I thought it was fantastic. I really enjoyed listening to all that stuff. Yeah. Mm-hmm. And someone wrote on the website that they loved the little conversation on sylvatic viruses. Mm-hmm. And I always have enjoyed this idea that, there's a cycle of arboviruses among, say, primates in the forest. And then there's a separate cycle in cities, like yellow fever and dengue in West Nile. So, yeah, that was fun. It was a good episode. We have a few more episodes stored up. In fact, I hate to tell you in a way, but in the next 2 weeks, we're not going to record because I'm away. And we'll release those, but hopefully you'll enjoy them. But today we're going to talk about coronaviruses. The first thing I want to discuss with my colleagues here, we have a new name for the disease, COVID-19, and the virus is SARS-CoV-2. Any thoughts? Do we like it?
Well, my first thought is, why did we have to have 2 names? Because for those of us that are pedantic, every time we hear the virus referred to as COVID-19, we cringe because that's the disease. So that was one reaction. And it's like having AIDS and HIV-1. We have the disease and we have the virus. But I don't think we needed to have a name for the disease myself.
Because with SARS, the original SARS, the disease was built into the virus, right?
Right.
SARS, severe acute respiratory syndrome coronavirus.
Right.
So why couldn't they name this virus SARS-CoV-2? You got it. The disease is right there. And as someone said, now, wait a minute. We have SARS-CoV-2 causing not SARS, but COVID.
Yeah.
And COVID is what? Coronavirus infectious disease? Is that right?
I think that's what it is, which means that then when you say COVID-19 disease, it's redundant.
But also infectious disease is about as bland as you can get, right?
Yeah.
Yeah, it seems to me like, something more descriptive about acute respiratory distress. But then, of course, you're back to SARS. It's tough. I also have, first of all, I don't care. But if I'm going to jump on the pedantic bandwagon, I have a a sort of a reaction to SARS-CoV-2, because to me, SARS is SARS. Okay. So now SARS is something different than it used to be. And what's SARS? Is SARS-CoV-1? Has somebody done that?
Yeah. Are we going to have to distinguish that
by implication? Yeah.
Apparently, that's not going to change.
Oh.
It's just going to be SARS-CoV.
Okay.
But we'll leave that up to the ICTV, right?
Yeah.
I don't know. Anyway, I don't terribly care, but the press will call the virus COVID-19, and although most people don't care, people call it the AIDS virus all the time when we have a name for the virus, right? HIV-1.
Yeah, sure.
So anyway, that's that. But now, from now on, we shall use those terms.
We shall try to use those terms. I mean, maybe you can do it. I'm going to have trouble.
What are you going to do?
I don't know. We'll find out.
We'll see.
We'll figure it out.
Is that Dickson?
Yeah,
It
here.
is. Hey, how you doing there, guys?
What happened to your course? It ended early?
Well, we had a review for the midterm, so I got out early.
You got the midterm is next week?
Yeah.
Not much of a review, huh?
They didn't want one. I told them to review the PowerPoint slides. That's what I told them to do.
All right, so we let's take a look at the Johns Hopkins dashboard, which has a now at, and I think Rich pointed this out, now has in the graph, it has a daily increase.
Yeah,
finally. Which we were asking for, and I doubt they heard us, but I'm glad they responded.
I see this sort of presentation referred to in other sources as an epidemic curve.
Yeah, that's exactly what it is. Yeah.
Which to me is what this is a report on a daily basis of new cases or new recovered or whatever. In this case, they present new confirmed cases and new recovered cases, so that you can. I think it's much easier to see what's happening with respect to time, what's
for sure.
There's two things impressive about this presentation at this point that I'm sure we'll discuss in some more detail. But number one, the new cases have more or less been decreasing with respect to time, which would mean, at least since 90% of these are coming from mainland China, so would suggest that it's on the wane, at least in China. I'm being careful about this because I don't want to say it's over, because probably not. And the other thing that's striking about this is that on February 13th, there was a huge, an apparent huge spike in cases. And people are probably aware of this, but it's worth pointing out that what happened was that the Chinese decided that they were going to redefine what they called a confirmed case and include cases that they were calling, what am I going to call it now, COVID-19, that they were calling COVID-19 based on a clinical diagnosis, which could include, it doesn't have to include PCR or some sort of molecular diagnosis that confirms the presence of the virus, but rather just things like a CT scan that shows what is apparently a viral pneumonia. And so using that new classification, they picked up a whole bunch of cases that looked like the disease, but had not been confirmed using molecular techniques. Personally, I'm not crazy about that idea, because that winds up including things like, for example, other viral pneumonias, for example, flu and et cetera. And now, as of yesterday, I guess, they've decided that was a bad idea. And they're back now to doing cases that are just confirmed molecularly. And so this is going to cause some wonkiness in these curves for a little while till it stabilizes.
My understanding is they're going to go back and retest some of those.
Yeah,
right?
Exactly.
So I just want to point out, if people are looking at the dashboard, the curve in the bottom right, by default, I think usually comes up as actual, but there's 2 tabs underneath. You can click on logarithmic and daily increase. And so it's that daily increase where on February 12th, the new confirmed cases 419, and on the 13th, 15,100.
Exactly.
And then as of February 21st, back to 599.
It's a mess.
So as of today, we have 76,798 total confirmed globally. Most of those in mainland China, because 634 outside of China.
Right.
Most of them in China. And the 634 went up because of the cruise ship recently.
Yeah.
But again, really pretty good job containing this within China. And we have 2,250 deaths. That's gone up a bit since we spoke last time, right?
Dude.
Right. So just taking a straight ratio of that, the 2,200 deaths over the 77,000 confirmed cases, that comes out to 2.9%.
Exactly.
But as we will probably discuss, that's a very squishy number.
Yeah, I think last time we talked about this idea of doing that math just the way you just did it, right? Mm-hmm. That first of all, it's early on still, and we don't have all the right numbers, right? We don't know. There may be many, many more cases without fatalities, which would bring it down. It's just, I don't know, if we're going to get an eventual number. I think at some point when this is over, they'll do serological surveys and really see how many people were infected by antibodies. And then you can ask, what is the CFR? Because right now it's not accurate because we don't really know how many cases there are.
And we don't have a good so we don't have a good numerator either because of what we just described, deaths and what are those due to, and the denominator, how many total cases are there? We don't have that really.
Yeah. So this
go ahead. Sorry.
Go ahead.
Sorry, just so the people have another presentation in their heads. If you take the ratio of the cases outside mainland China to inside mainland China, you come up with a number that 98% of the confirmed cases are in mainland China. So there's only 2% of the total that's outside China.
Right.
And within China, it varies also from north to south. Right.
Mm-hmm.
So
Right.
in the Wuhan area, it's very high. It's very high because the system is overwhelmed. There are not enough hospital beds for everyone. So they go to lower skilled care centers and there you may die. Whereas in the south, they're not overwhelmed. They can take care of it. So it has, like the last time we talked, 0.1% fatality rate versus.
Does this mean that if you get the proper medical care early on, you're not going to die from this?
Well, that's always the case. Even with Ebola, if you get proper care early on, the CFR is.
It's not true for
different.
pneumonia among old people.
Well, among old, so age is another factor that if you do the CFR in age, it goes way up. In fact, one of the papers we'll look at in a moment shows that it's like in people your age, Dickson, it's like 14%.
I know.
You haven't gone.
Yeah, Dickson, you better just
chill.
Say your prayers.
Well, you should just.
Goodbye, Dickson.
I do every night.
So I want to say, I was in NIH yesterday and the day before for a meeting on a virus which will probably rear its head in the fall, enterovirus 68.
Oh, yeah.
But I was talking to Ian Lipkin, and he said that the diagnostic situation in China is not good. They're having trouble with their PCR, and he wanted to send someone over to help him out. He's got an interesting PCR he's developed here for it, so it's problematic. And now a number of people have written in, and we'll see. They say, "Why are you depending on the numbers out of China?" And like my response is,
"What else you got?
What else do we have?"
That's exactly right.
You could speculate all you want. We know it's an underestimate,
right?
And there's nothing we can do because it's them doing the tests. We cannot walk in there and start doing tests.
So essentially, this looks exactly like it did with SARS for me.
In what way?
In the chaotic aftermath of the epidemic. It's just chaotic. There is no true public health service in China.
I'm not sure about that.
No, there's not.
The CDC has a dual function.
Yeah, I know.
So it's like ours, CDC plus hospital,
It's failing.
I don't know if it's failing. I think they're containing it from what we look like here, right?
Yeah, well, from what you look like. Because exactly.
That's not really well said from the numbers, from the daily numbers.
I know what you're saying.
Yeah, but listeners may think I'm incoherent, but I'm
actually you're quite coherent.
Actually, this is an aspect of this whole thing that interests me because given that 98% of these cases are in mainland China, then most of the infections and disease we're looking at comes from mainland China, and it does appear that the daily case rate is on the decline. And in fact, if I'm not mistaken, that the peak corresponds loosely with when China started to institute travel bans and quarantine procedures and that kind of stuff. And so I really wonder what it would look like without the measures that they have taken.
Sure.
And how, I mean, you can't do the control.
It would probably be worse.
Is this thing, are those procedures actually effective, or is this thing just burning out on its own?
Great.
I don't know.
You know what? They asked the same question when Jon Snow knocked the pump off in England to prevent the outbreak of cholera.
Yes, that's
and
true.
he knocked it off long after the epidemic was finished. So he's not given credit for actually quelling the epidemic. He's given credit for pointing out what it was due to.
Do you know that Ian Lipkin is the Jon Snow professor?
Yeah, I do.
For knocking a handle off and you're not even sure it did anything.
Exactly. It did nothing because the epidemic was over by then, but it said, don't drink this water ever again.
I think it's still important, right?
Yep. Very much
identifying
important.
a common cause.
Very much important.
So, Rich, you wrote in here this infection fatality rate. What does that? Explain that to us.
So this is from the WHO situation report from the 19th of February. I think it's report number 30. And I forget whether it's repeated later, but it doesn't matter. From February 19th. So they got a bunch of epidemiologists and mathematicians together to try and get estimates of an incubation period, a case fatality rate, and an infection fatality rate.
Mm-hmm.
Right.
And what they came up with was an incubation period of 5 to 6 days. And it's important. It's important that these are all very rough estimates for all the reasons we've already discussed. They came up with an incubation period of 5 to 6 days with a range of 0 to 14 days, a case fatality rate of 2.3%, and an infection fatality rate of anywhere from 0.3 to 1%. And the infection fatality rate tries to use as the denominator in this equation some estimate of what the actual number of infections are, including, people who aren't tested and et cetera. So this is a very squishy number. And there was an update in the February 20th report that refers to one group saying that they think they've nailed it down a little closer and have it at 0.94%. Okay, so that's still within the range of, it's a lot higher than 0.3%, but it's still within the range of 0.3 to 1%. So I get, if you do more math on that, that's saying that there's somewhere between twice as many and 8 times as many infections out there as have been confirmed. Okay.
This incubation period is important because that, of course, affects how long someone would be quarantined if they have a potential contact. And so currently it's 14 days, right?
Mm-hmm. Which is the sort of on the very long end of the estimates of the incubation period.
That's right.
Correct.
Now, Oliver sent us a really interesting article, the largest data set published so far from China on the virus, which at the time was 72,314 patient records, which is pretty close to what we are now.
Yeah, as of February 11th, 2020. So a little over a week ago.
And he says although the confirmed cases are 50-50 male-female, the fatalities are 2/3 male and 1/3 female.
Smokers.
It may turn out to be related to the prevalence of smoking among Chinese men versus women. In 2010, 54% of Chinese men smoked versus 2.6% of Chinese women.
That's incredible.
Wow. That is amazing.
That's incredible in a couple of ways. The difference is incredible, and it's incredible that over 50% of the men smoke.
No, that's 2010. It could have changed substantially in the last 10 years, right? So this is a huge number of cases, and let me just summarize some of these.
This is a great paper. This one
from the novel coronavirus pneumonia emergency response epidemiology team, and it's published in the China CDC Weekly. So again, over 72,000 patient records, 889 asymptomatic cases. Okay, a symptom is what you feel. I feel fever. I feel stomach ache, bone ache, muscle ache. Okay. These people didn't
fuzzy teeth.
Fuzzy teeth. They didn't feel any of that. So right now I feel great. Right. I have no symptoms.
Right.
There is such a thing in science as asymptomatic. And I could have a virus replicating in me. And if you measured my serum, you might detect cytokines or even low levels of RNA. I wouldn't know it.
So
I guarantee that you got a virus replicating.
I do.
Did you go through the source of the virus could also be in fecal?
We didn't get to that
events.
Yet. We'll get to it.
All right.
We'll get to it. Anyway, among confirmed cases, most were 86.6% between 30 and 79 years of age. Most were in Hubei. And listen to this, 80.9% were considered mild.
Sure.
And again, they say a case fatality rate of 2.3%, which is close to what Rich said before. And they say it took 30 days from this epidemic to spread from Hubei to the rest of China. And they say with many people returning after a long holiday, China needs to prepare for a possible rebound of the epidemic because they're already reporting here that it has started to decline. So I don't know what the timing is over there, but I guess they're coming back from, that nobody's traveling, right? So I don't know what's going on.
So under the age of 60, how many people died?
You'd have to add up the columns here. Let's take a look. Deaths. Let's do it by age. We can do that on the
that's
under the age of 60.
Oh yeah, they have it here.
Okay, so 130 between 50 and 59, 130. 40 to 49, 38, 30 to 39, 18, 20 to 29, 7, 10 to 19, 1, 0 to 9, 0. And there were 416 cases in that 0 to 9. So, just getting up above 60-ish here is the most.
They give, I think this is a very interesting question, Dickson, and generally, the answer is that the percentage of people who are infected who die is like so far 0 for people less than 9 years old, and then increases steadily up through 79, 80.
Oh yeah, look at that. We got paid.
So the
percent.
So the percent deaths in 70 to 79 is like 30%. Mm-hmm. In contrast to, a similar number of cases in 20 to 29-year-olds is 0.7%.
Yeah.
So that's more than 30 times more in people who are 70 to 79. So it's a striking trend.
So Dickson, the overall CFR is 2.3%.
Right.
Yeah, below 50, it's lower than that. But now in the 60 to 69, it's 3.6%. 70 to 79, it's 8%.
Right.
Over 80, 15%.
Exactly.
All right. So CFR can be influenced by a lot of things.
So how does this match with the WHO statistics on influenza deaths? Were these the same cohorts?
I don't know if stratified by age. I don't have that with me right now. That would be very interesting. All I know is in the U.S. right now, there have been, 8,000 deaths out of 15 million cases.
Exactly.
Yeah. It's lower CFR overall, but I don't know how the stratification is very important.
Age is still
I totally agree. I don't know the answer to that.
Okay, fine.
But this 15% in the over 80 is.
Do you learn anything from these statistics with regards to the immune mechanisms at work here? Does something fade rapidly when you get to be 60 years old? We were afraid of your T cells not there anymore, right?
Your immune system fades. So, I don't have any bone marrow.
That's right. It's gone. What do you
long
think?
Bones, anyway.
How do you think I feel like?
I felt like a shell of a human. I said
hollow bone. That's what they're going to call me, old hollow bone.
No, as you age, not only does your immune system decline, and so you don't make any new memory T cells,
That's right. That's right.
And your breath gets shorter. You don't have the muscles for exhaling anymore.
So it raises a lot of interesting questions about potential intervention strategies. Namely, when you're 50 years old, why don't they take a bone marrow sample and then freeze it? And then when you get to be 70 and you get one of these diseases, give you the bone marrow back.
That's another podcast. It's a great question. I don't know the answers.
So the another section of this that I found fascinating is the correlation with comorbidities.
Right.
Okay. So the case fatality rate amongst people with no other issues.
Right.
The case fatality rate is 0.9%.
That's right.
Whereas, for example, the biggest one is people with cardiovascular disease. It's 10%.
Sure.
So if you're, it pays to be healthy.
But they say here, listen, Dickson and everyone, in light of this rapid spread, it is fortunate that the illness has been mild for 81% of patients. All right, so this is a very low over case fatality. So this is to tell people, don't worry so much.
I'm concerned about certain news releases that go against this statistic. Basically, you had a house full of people of all ages that died from this. They all died.
But if you look in the numbers, there are exceptions.
No. I know that, but an entire household of people.
Well, in China?
Yeah.
Well, it's in these numbers. It's in the 72,000.
No, but isn't... All right.
I don't know what the reason for that is.
What about... Well, that's...
But it doesn't happen in every household, obviously, because 80% are mild.
No, that's what I'm saying. That's just exactly
well, the news likes to focus on one deadly incident like that. And that's not right.
They focus on anecdotes instead of overall statistics.
Understood.
We try not to.
Yeah.
We try to look at the data like we're doing here in this paper.
Sometimes the exceptions are the biggest stories in terms of what's really going on.
I understand. Is that true for science, Dickson?
Always. It's not true for scientists, but it's true for science.
I want to also point out, they say a major contribution of our study is a first description of the COVID-19 epidemic curves. And that is, the daily number of cases, as Rich said. And they say, we interpret the overall curve as having a mixed outbreak pattern. The data appeared to indicate a continuous common source pattern of spread in December, and then from early January through February 11th, then the data appear to have a propagated source pattern.
Exactly.
So let's say what those are.
This sounds just like cholera. But
a mixed outbreak pattern where you're having seeding, in this case, zoonotic seeding at multiple places, right?
Yep.
You get an outbreak here. Not a continuous common source where you have a source that infects others, they infect others, right? Exactly. Chains of infection.
Exactly.
So they think that there were several zoonotic events early on. Mm-hmm. They say the fish market, but I'm not sure that it was there. Maybe it was there, and plus other places too. Yeah. And then later on, it started to propagate serially. That's good. They also offer the first description of the 1,700 confirmed cases among health workers.
Right.
Which happened, unfortunately. But they said no evidence for super spreaders among those.
And of those people, how many of them died?
Here we go. It's table number 2. Case fatality rates.
Because these are usually
China overall
30 to 40 age people.
It looks like, well, it depends when, they do it for several periods. It's 5% between January 1st and 10, 0.3% January 11th to 20, but this is based on 1 or 2 deaths.
Right.
There are only 5, 6, 7, 8, 9, 10 deaths.
Right. Out of 1,400 people.
1,700, so it's very low.
17. Very.
Well, if you're... Go ahead.
No, I was just going to say, can we go back to the statement? So you described what a mixed outbreak pattern is.
Right.
And so the overall having mixed outbreak, but a continuous common source pattern in December. So continuous common source pattern, we understand what that means? We've defined that for the audience?
So mixed outbreak. Oh, that means mixed outbreak means two patterns. The first is continuous common source.
Right.
Where there's one source that's spreading multiple times.
Okay, and that was in
December. Right. And then
from early January to February, a propagated source pattern. So what do we mean by a propagated source?
So there you have a person infected, and then that person infects another and another, chains of transmission from it's sort of like your first case. What's the word for that? That's escaping me. The first case,
like patient zero.
Patient zero, exactly. That would be from patient zero onwards would be a propagated source. Whereas the mixed outbreak, you have continuous common source. You have a source infecting multiple people, and it's not chains of transmission yet.
Like the pump in London.
Yeah. That's what you said before, Dickson. It's like cholera, right?
I did say that, actually.
Okay.
So mixed, I didn't actually do this right in the first. Thank you for bringing us back. The mixed outbreak refers to the two, the common source and then the propagated source.
So you can say point source and non-point source, and it's still valid, and that's a clearer way of looking at it, I guess.
Okay.
Anyway, the thing I wanted to say was if you're a healthcare worker and you get sick, you're more likely to get care right away, right?
You are.
Like the day of illness, you're going to go, hey, I don't feel well, here, go home, or we'll take your temperature and do that.
You recall the Lassa fever outbreak to begin with back in the 60s.
Yeah.
Only healthcare workers were died. There was one person admitted and 3 people that took care of him died.
Yeah.
So you had 1 to 3. That was horrible.
If you're a healthcare worker, it seems to me you're probably more likely to be not 70 years old with a comorbid condition.
No, you're right. You're absolutely right, too. That's true.
Just 2 more things from this I wanted to bring up. One, so at the end, they praised China for containing this outbreak. All right, that's good. That's fine. And they do say with our other international partners, thank you very much.
Like United Airlines.
And then they say the coronavirus and others may continue to adapt over time to become more virulent. And I don't know where more virulent comes out of this. We don't see any evidence of increased virulence.
That's ridiculous.
No.
They do say zoonosis is not going to stop, which is absolutely true.
Okay, big question here. Have they been tracking the actual sequence of the virus?
Yeah,
and how has that been varying?
It's all the same more or less. There are variations, of course, but if you look at the phylogenetic tree, there's an ancestor to all of them. There's nothing breaking off, splintering. In fact, we have a link from someone coming up where there's a nice website where you can see real-time tracking of the sequences.
So if the virulence was increasing, there would be some correlation with the sequence.
There should be.
Well.
There should be a branch off the tree. But also in terms of the disease, we don't see any evidence for, a temporal increase in virulence. I don't know why you would expect that anyway. Maybe an increase in transmission, but I think that happened early on in the epidemic, right?
Yeah,
so we'll actually talk about that. So I don't know why people say this all the time. Why increase in virulence? I don't know. It doesn't happen.
It's a fear of the unknown, probably.
Okay. All right. So, Kathy, you want to tell us about these things you put in here?
Let me get. So this is the paper that you were just talking about. Also shows we didn't discuss it, but there's some difference in sex of the people. Okay, and so I'm desperately trying to pull up my Google Doc to.
In this case, we thought it was because of smoking, right?
Right.
Because men smoke and women don't, basically, in China. Or maybe that's part of it, right?
So monkeys lie and...
It could be part of it. But I just wanted to point out that there was an article today in the New York Times quoting Sabra Klein about sex differences in infectious disease in general, and particularly citing a paper from Stanley Perlman, who we had on the TWiV when we were in Iowa last March, where Stanley did a study with male and female mice and SARS, and they have evidence that there was a difference between the infections in the mice. The male mice developed SARS at lower viral exposures, had a lower immune response, and were slower to clear the virus from their bodies they had more lung damage and died at higher rates.
Just lazy. That's it.
With a lot of parasitic infections, these differences are quite obvious. And it relates primarily to the menstrual cycle of the female animals.
Right. So I was going to go on and say when they blocked estrogen in the infected females or removed their ovaries, they were more likely to die. But blocking testosterone in male mice made no difference.
Right.
So that indicates that estrogen may play a protective role.
Indeed.
And as Sabra said, this plays out in a lot of infectious diseases. And the downside for women is that this immune response can lead to more things like lupus and autoimmune diseases, rheumatoid arthritis and lupus. So this is just kind of playing out here for this.
So the idea is that the women have generally a more robust immune system, but that's not all good news, right?
Right.
Because it can go
Exactly.
wrong.
Right.
Okay.
Did that article also talk about behavioral differences?
The New York Times article?
Yeah.
No.
Yeah.
No,
Too
it's
bad.
just replicating. I don't. At least.
No, actually, I
remember.
think towards the end it does. It talks about environmental differences and et cetera, and mentions smoking.
A lot of a lot of the differences,
Type 2 diabetes, blood pressure.
Yes, you're right.
a lot of differences between male and female acquisition of infection, let's say schistosomiasis or malaria, relates to their work habits.
Mm-hmm.
And if you're not outside a lot, then you're less likely to encounter these infections than if you are. And so it breaks down into either females or males are out more than each other. And that's the reason why that difference exists. It's just behavioral. So there's that aspect to it. That's all.
I want to point out a paper, Journal of Medical Virology, it's a commentary, I guess. Does SARS coronavirus 2 have a longer incubation period than SARS and MERS? And they looked at data for all of them and tried to get as much data in well-defined exposure periods. And the answer is SARS coronavirus 2, 4.9 days with a range of 4.4 to 5.5. SARS 4.7, 4.3 to 5.1 range, and MERS 5.8, 5 to 6.5. And these are all the same. So they're basically all the same. About, the mean is 5 days for SARS coronavirus. Yeah. So I don't know, 14 days. You think that's good?
That's 2 standard deviations beyond the mean, sounds like to me.
What was the range again? Did I say the
0
range?
to 14. The range was in the paper. I don't know.
It's 4.5 to 5.5. That's interesting.
So didn't they also show that some people who had recovered from the infection and been released could also be shedding the virus?
Yeah. I don't know where that is, so I don't want to do anecdotal news reports.
No, it was in the finding the virus in the feces.
Well,
I read that somewhere along the line here.
I don't know. Maybe we'll get to it.
I hope so.
But if they find PCR product, I don't care.
They have to do a plaque assay, of course.
Well, we're just getting infectious virus because PCR,
no, you're right.
Broken pieces of viral genomes cannot transmit infection.
You're absolutely
Unless you put enough of them in a cell which in the old days we used to call marker rescue, I think.
Vincent, if I've learned nothing on this show for the last
Kathy
300
and I
years.
Condit know that.
I subscribe to that hypothesis.
And the 2 weeks ago we talked about the sewage and transmission, and it turned out that it's happening here. So
that's right.
Not that we're prescient, it's just that you look back. So at this meeting in Washington, I gave a talk called Lessons from Polio for EVD68 and Acute Flaccid Myelitis. My last slide was George Santayana. Those who forget history are condemned to repeat it. I said it applies to science too.
No question.
How many of you in the audience read a paper that's over 2 years old?
I wait for them to grow up and then I read them.
All right. So there's a very brief article in Nature, which I wanted to mention. It's called More Than 80 Clinical Trials Launched to Test Coronavirus Treatments. And it's kind of driving WHO crazy because it's a mix of real antivirals and traditional Chinese medicines. And, the WHO would like these to be properly designed and controlled, but it's not always the case. But I suppose we'll see some of these results
What about,
at some point.
do they also cover convalescent serum treatment?
Yes.
Yeah, I'm sure they do.
Mm-hmm.
And what did they see?
They're in progress.
They're all in progress.
Yeah, all in progress.
So, they have, they're using other antivirals. So they're HIV protease inhibitors, loprinavir and ritonavir. They've been tested in SARS and MERS before, so they're being tested now. Remdesivir, a nucleotide analog, is being tested. It's been shown previously to work on other coronaviruses. Yeah.
Dickson, they have a 300-person controlled trial to test serum from COVID-19 survivors.
Right.
So.
Okay.
Yeah. And then they're testing chloroquine.
Really?
Which they say a malaria drug that killed off the new coronavirus in cell culture. I hate that word, killed off.
Yeah, exactly.
I mean, this is nature.
I know.
Yeah.
I know.
An interesting thing is the picture at the lead part of this story shows these people in their PPE, and then they have these big Chinese characters written on their PPE.
Right.
And I'm wondering what that is, if that's their name because they don't have name tags or what it is.
Do not touch this person.
Right.
PPE, personal
protection.
On their lab coats, yeah.
Personal protective, what E?
Equipment.
Equipment. Right.
Equipment, okay.
PPE.
Yeah.
They're also doing stem cell trials, which they're a little skeptical about.
Oh, boy, I'll say.
And then traditional Chinese medicines, including
pangolins.
Shang Huang Lian, a Chinese herbal medicine that contains extracts from the dried fruit lian shao. Forsythia fructus. I can do the Latin. That's fine. Mm-hmm. Okay, so anyway, it's all in progress, and we'll talk about it when they come out.
Right. I get questions from people all the time. Well, are they testing for anything to treat the disease? The answer is yes. Big time.
That's right.
But we don't know anything.
That's right.
And we will talk about it when we get the papers, because I don't want to do the anecdotal stuff.
Yep.
I want to talk a little bit about the genomes of these viruses and pangolins. And a very nice paper came out in Virological. I'm not sure it's a journal. Is it Virological?
Is this the proximal origin of SARS-CoV-2, that paper? I couldn't quite figure out what it's in either.
Yeah. Anyway, it's a paper by Christian Anderson, Andrew Rambeau, Ian Lipkin, Eddie Holmes, and Robert Gary.
Wow.
It's called The Proximal Origin of SARS-CoV-2, which I really like that, proximal origin.
Mm-hmm.
And it covers stuff that we missed last week. So let's talk about it a little. This is very cool. And there are two important things here. So they say in the first paragraph, the sequence that we have now from the virus in humans, the closest virus in bats from the 2013 bat, and then the new isolates from pangolins, some of their features provide evidence, this is their saying, that SARS-CoV-2 is not a laboratory construct nor a purposefully manipulated virus. And us on the TWiV team were kind of angsting over this last week. My fault, because I said, hey, did you hear this? And I tell you, it really bothered me that there was some suggestion that this was a lab construct. Because if it turned out to be true, that would bother the hell out of me. Not just because of people dying and so forth, but it's kind of an indictment of the field, right?
Mm-hmm.
Right.
So the genome, there are two things about the spike, right? These are envelope viruses that have a spike glycoprotein embedded in the envelope. It's called spike, or S protein, which attaches to cell receptors and then catalyzes fusion of the viral and cell membranes. And so there are two aspects of that spike that really bear light or shed light on the origin of this virus. The first is the receptor binding domain, and the second is a furin cleavage site. So the spike is cool because there is a very distinct and separate fold of the protein called the receptor binding domain that it by itself will bind the receptor. And in this case, the receptor for SARS-CoV-2 is the protein ACE2, angiotensin converting enzyme 2 on the cell surface. And it was shown some while ago that this virus binds with quite high affinity to ACE2. Now, the ribosome binding domain there are 6 amino acid residues within that are
the receptor binding domain. You said ribosomes.
Right.
Yeah, sorry. Good thing I have people listening, right?
Yeah.
Receptor binding domain, RBD. 6 amino acids are critical. If you change them, you lower the affinity of binding, and they all make contact with the receptor, so they're really important. Now, when you look at this this sequence, these 6 amino acids, the human virus, 5 out of 6 of those residues are identical in the bad isolate from 2013. Okay, so that's point number 1. The second is when you look at these residues and you take into account all the known coronaviruses that can bind ACE2, and you look at all their sequences, and you do a computational analysis, you look at this one in the SARS-CoV-2, it says the computer says it's not a very good binder based on what we know. But when the actual biochemical experiments were done, it turns out to bind with high affinity. And so that's the first bit of evidence. It looks like this SARS-CoV-2 was actually, this particular sequence of these 6 amino acids was derived by selection in cells. Because if you wanted to engineer an RBD in this virus that could bind ACE2, you would have used something we knew bind well, not something you didn't know bind well, right?
Right.
Yeah.
So that, I think, to me, that's quite compelling.
Yeah, I really like that, and it's it's what we've said so many times before in discussing this notion of creating agents, viruses in the laboratory that would wreak havoc in the general population, is that nature is much slicker at doing this than us. We couldn't have imagined that changing those residues in that way would give a good binder based on all of the information we have, but nature figured it out.
So exactly where this came from, we'll talk about in a moment. So, but this RBD is one important feature. Then the other feature in the spike glycoprotein is a cleavage site for proteases called furins. And it consists of an amino acid sequence arg, alanine, arginine. And the thing about the spike is it has to be cleaved in order for fusion to occur. And the cleavage is done by proteases, typically cell proteases. And that, of course, depending on whether the cell has it or not, that can regulate the infectivity.
And that brings the envelope closer to the host cell membrane?
It brings the fusion protein close to the cell membrane so it can insert and catalyze fusion. So the really interesting thing that was seen a couple of weeks ago was that the spike of Corona-2, SARS coronavirus 2, has a furin cleavage site between the 2 domains that are known to be cleaved. And that's not present in the bad isolate, right? And furin is interesting for a number of reasons. First of all, it's in most animals, but it's also highly produced in the lungs, the respiratory tract. And we know that for influenza virus, if avian influenza viruses pass through chickens, they can acquire a furin cleavage site, and it makes them more transmissible and more pathogenic. And then last year, Ralph Barrick's lab showed that if you take some SARS corona-like viruses from bats and you treat them with trypsin to cleave the spike, it enhances their infection of human cells. So the idea is that this furin cleavage site is essential, and we don't know this because it hasn't been shown yet, but I'm sure it will be. It's essential for infecting human cells. All right, so the experiment is to take that bad virus without the furin cleavage site, but with a high affinity RBD for ACE2 and see if there's a difference in infecting cells. So that's interesting. But the other thing that came with the furin cleavage site is there's a proline just before this RRAR. So it's actually PRRA. And so they say the proline actually allows the addition of O-linked glycans, sugars. And furthermore, these sugars are typically introduced in response to immune selection. So again, let's say a human wanted to put a furin cleavage site here. They wouldn't have put the proline upstream.
Okay. Right.
What? You don't need it for.
I would have.
the proline was selected in some animal by the immune response and then the glycans act as a shield. The sugars act as a shield against the immune response. So that's why the furin was most likely not inserted in there by a human. So those are the two things, the RBD and the furin. They are new to this virus. And in fact, this RBD was not found in any other similar virus from bats. So where did these come from? There are a couple of theories which are kind of interesting. And one is that these viruses circulate in animals, and somewhere in some animal with a human ACE2-like receptor that's selected for a high affinity RBD. And those are circulating in animals, and then it acquired a furin site because there are furin sites in other coronaviruses. And there you now have a virus that it can infect human, and all it has to do is get into people. And, where that happened and when, we don't know. But this virus could have arisen 1,000 years ago or a year ago or a month ago, and then maybe it's brought into market and it starts this outbreak. So the first idea is it was selected in animals, but the other is that maybe these viruses have been repeatedly infecting humans, and then at one point the furin site got in and it took off, right? And so I like that because let's say there have been sporadic zoonotic spillovers. You get a case of pneumonia here and there. Nobody notices. You get pneumonia in the winter, most of it not diagnosed, right? So you wouldn't know. And then all of a sudden the furin site gets in by mutation or recombination. We don't really know. And now it has the ability to transmit effectively among people. And they point out that this is the way MERS is working now. There are short chains of infection from camels, but then they fizzle out because that virus has not yet acquired the ability to transmit effectively among humans, where, of course, this new coronavirus has. So I think they need to do a lot of surveillance in animals to try and see if they can pick up any of this. And they say we should do sero surveillance after the epidemic and see if we can differentiate between this particular corona and others that may have infected people at other times. I think it'll be fascinating to try and it's kind of like a detective story, right?
So this brings up in my mind, of course, takes me back to all of the stuff about transmission of bird flu, okay, where they have a model for transmission in ferrets, and, it makes me want to do the experiments with this virus of looking at with or without the furin site. Is it how does that affect transmission?
What would you do it in? What would the animal?
Well, that's my question. Is there an appropriate animal model to look at transmission for
try a pangolin.
Coronaviruses? I don't know.
Pangolins are protected.
Yeah, they say that, but they're still eating them. And they're importing them from Africa now.
I have a lot to say about pangolins.
Rich, for SARS, they use a mouse-adapted SARS virus with, and I believe they put the receptor into mice, but I'm not sure offhand. I think Ralph told us that last time. Ralph will be back in 2 weeks, by the way. And
okay, good. And you can let's write this question down for him. So that's a model where you can measure transmission.
I don't know if you. No, I don't think you can do transmission in that. Yeah, that's a good question. How can we study transmission? Because those are really essential experiments. So where does the pangolin come in? So pangolins are regulated. It's illegal to traffic in pangolins in China. However, they're the most illegally trafficked mammal in the world.
Exactly.
And they come into China from Malaysia, probably from other places too. But
Africa too, though.
The sources in this case, they capture these in the south of China, in the Guangdong province. They capture them, and they have some stored. So they went back and looked at them, and they found coronavirus sequences in them, highly related to this virus. And in fact, they could recover entire genomes from pangolins, mainly from the respiratory tract, implying a respiratory disease in pangolins. We're not clear on that. And there seem to be two lineages, but they're not as close in genome sequence to the human virus as is the bat virus that is circulating that was picked up in the 2013. I pasted in a phylogenetic tree so you can see that. It's the Wuhan virus is really close to this bat virus called Rat G13 2013. And then we have Manis javanica, which is the pangolin. They're close, but they're not as close as the bat virus. So this pangolin isolate is not the immediate, the proximal ancestor of coronavirus 2.
However, it's got the cool receptor binding.
The receptor binding domain is 6. Those key 6 amino acids, all 6 are identical.
That we could never have dreamed up. It's there, and that means that you could come up with those 6 amino acid sequence by passage in a animal with a human ACE-like 2 protein, and animals have human ACE-like 2 proteins in them. So, this is selected for somewhere. So that's part of the puzzle that you can select for this high affinity RBD, and then of course the other part is the furin site, which is not found in the pangolin isolates. There's no furin cleavage site. So if that was, these viruses are clearly circulating in various animals, and they may have high affinity RBDs, but they need to acquire a furin site. So they need to keep looking in other animals. This is not it. However, the attention paid to pangolins in the past couple of weeks may be good. Maybe they'll stop trafficking them. They're really nice looking animals. Yeah.
Oh yeah.
I just
really
cool.
Put in this Nat Geo thing of the what's it called here? 12 photos show the adorable pangolin in all its glory. And in one of them, they show one of the caregivers helping the pangolin dig for ants. And they say specifically that the caregiver is not named for his safety to help prevent him from being targeted by traffickers who want access to the rescued pangolins.
Exactly.
It's really nice. But, I saw a movie where they were just throwing them in sacks. They were catching them out there, throwing them in sacks and shipping them. It's sad,
So I put in a link to the wiki article on them, and it's interesting because they are these things that kind of, sort of, maybe kind of look like anteaters and get associated with anteaters because they have a long tongue and they eat ants. Okay, and they're kind of the same sort of shape. It turns out,
and termites too,
that they are not in that same family. They're in I don't know if it's a clade called Ferae, which is a clade of mammals, and they're split off. One of the closest relatives is carnivores: cats, hyenas, dogs, bears, seals, and they're these cute little creatures with these keratin scales all over them that they use for defense, and they are prized both for their meat, so their food, but the scales are used in Chinese traditional medicine, and so that's why they get trafficked so much. And I think that that's a tragedy. I think I'm sorry that they aren't the just proximal source of this virus and absolutely chock full of it. Okay. Yeah. So that people would then just leave them alone.
Yeah.
Well, that means all the traffickers would die from it.
Good idea.
Exactly.
So, and they curl up in a ball when they feel threatened. And so when you were talking about what they're closely related to, I was looking at that picture thinking,
hedgehogs.
Pine cones. That's what they look like, pine cones.
No, you're absolutely right.
There's a cute picture of, a child on top of a mother, and there's one in a hospital all bandaged up. Yeah, as only Nat Geo can do.
Those are great pictures.
Yeah.
So pangolins are the only mammals other than bats known to be infected with the new coronavirus so far. So we have to look some more. And, these came from Malaysia. So they should look in other, sources of pangolins, other animals. I'm sure they're going to do all this.
It's not the only place that they're eaten either. So that if that was true, that they would have had other outbreaks of this coronavirus in other places.
Let me summarize. The bat virus, the pangolin virus, they have this high affinity RBD, but they do not have the furin cleavage site. The original idea that this was a laboratory or associated virus was that it had a furin cleavage site, and they said, this must have been put in a laboratory. But for the reasons we said today, that's likely not true. But even if so, there's no virus that we know of close enough to this human to be the progenitor. So even if you took the bat virus and put in the furin site, it's too different. Right. There are not enough nucleotide differences that it wouldn't have been the precursor. And they point out in this proximal paper that, there's no known laboratory worked on corona that could have been the ancestor of this virus. They're just not close enough. So I know some people will not believe that still, but
that's the science.
It's the science that says it, right?
It's not a belief system.
Right. I did look up a little bit about where this was posted, and it's a site called virological.org, which is a discussion forum for analysis and interpretation of virus molecular evolution and epidemiology. The administrator is Andrew Rambeau, who's professor of molecular evolution in Edinburgh. And he tweeted 4 days ago, I've just posted a preprint of a review. So this is a preprint, not yet peer-reviewed, but there you go.
I think it's very good. It's accessible, so we'll put a link and you should check it out. It's really nice. Mm-hmm. Okay, let's do some email. I know everyone has sent in a lot of email, and we appreciate it. We'll try and answer them. I wanted to read this first one from Justin, who, this is a news report of a baby who tested positive for coronavirus 30 hours after birth. What I think is interesting is Justin's comment. No word on test method, though, i.e. PCR for an RNA virus doesn't mean there's active infection, just means there's a really good PCR machine somewhere. And I think that's perfect, right? Mm-hmm. I'm glad Justin understands this, that we don't know what it means.
Yeah.
And then Mark sent in a link to an old clinical trial of ritonavir and lopinavir, the HIV protease inhibitors, against SARS coronavirus a long time ago. But again, his comment was funny. Certain to warm the hearts of Vincent and listeners is that they've used plaque assays. Yes. All right. Rich, can you take that anonymous one?
Okay. So, as a matter of fact, this is a compilation of some correspondence that we've had with this individual. I'm catching up with the latest TWiV episode today and felt compelled to write in. The TWiV hosts commented on Dr. Zhengli Shi's statement that, quote, the novel 2019 coronavirus is nature punishing the human race for keeping uncivilized living habits, end quote. I certainly agree with the comment that nature really does not care about mankind punishing us. But I echo the feeling of Dr. Shi that this is perhaps punishment for bad human habits. Not nature doing it, but you can look at it this way. I believe what she referred to is the sale and consumption of wild animals in some parts of China, which could turn out to be a major factor for initiating current epidemics. As reported widely in the news media, pangolins are now considered to be the likely intermediate hosts. Well, no, there may be somewhere along the way, but it's okay. That was old news. Transmitting this coronavirus to humans. There's also a paper published in October last year that some Malayan pangolins illegally smuggled into China were infected with unknown coronaviruses and died from lung disease. If further studies ultimately confirm this pangolin connection, I hate to say that we indeed are collectively punished by some people's demand of pangolins as delicacy or medicine, and by authorities not doing enough to stop the illegal sale and trade of, and transport of pangolins. This could be an important lesson from nature for the importance of respecting nature and protecting animal habitats. Hear, hear. As a virologist of Chinese origin, I have been captivated with all the coronavirus-related news, including popular posts in the Chinese and social media. I have witnessed from the Chinese social media quite widespread acceptance of various conspiracy theories on the origin of the current coronavirus and the associated backlash against virus research in general. Even some of the highly educated friends of mine in China openly question whether the Wuhan coronavirus is man-made or leaked from the lab by accident. Dr. Shengli, she has been the public enemy number one in some Chinese social media circles because she happens to conduct coronavirus research in the city where the current epidemic started. I think the main reason for this phenomena is the public mistrust of the government and any entities connected with the government in China. Independent and scientific voices like TWiV could have been very valuable in China for defeating conspiracy theories. I'd love to hear what TWiV hosts think about the pangolin connection. Well, we've discussed this just above. Dial back in the podcast.
Right.
I myself am leaning towards the possibility that pangolins are dead-end hosts that have gotten the viruses from bats or some other animals. Even as dead-end hosts, they may still play a critical role in transmitting the virus to humans. Pangolin scales have been used in Chinese traditional medicines for over 1,000 years. This is one reason China is the number one destination for smuggled pangolins. Chinese traditional medicine is a highly controversial topic in China. Many Chinese people, including the government, promote the Chinese traditional medicines as a cultural heritage and believe in their healing power, even against the new SARS-CoV-2. Many other Chinese, including myself, consider the traditional Chinese medicines total BS. I think some of these traditional medicines do more harm than just being benign placebo. I speak out against the traditional Chinese medicines whenever I can, but many people just believe in the anecdotal evidence, and it's really hard to change their views. Yes, indeed. There is a widespread public fury over Chinese CDC and its scientists. Chinese CDC has the dual function of disease control like U.S. CDC and research and development like U.S. NIAID. Because the public was only informed about the human-to-human transmission of a Wuhan coronavirus sometime in January, while the Chinese CDC scientists published a retrospective report of early cases in the New England Journal of Medicine indicating human-to-human transmission in December last year, there is an accusation that the Chinese CDC scientists prioritized publications in international journals over disease control in China. The head of Chinese CDC, George Gao, has been the prime target of the public anger in the Chinese social media. The general public appears to demand Chinese CDC focus on disease control, not research or publications. This epidemic also brought the gain-of-function research and biosafety issues to the attention of the general public in China. The 2015 Nature Medicine paper on recombinant coronavirus from Ralph Barrick's group was widely distributed in Chinese social media, bringing a lot of fear over gain-of-function research. There's also a lot of concern over biosafety of virology research. Some people even suggest that virology research should not be performed in large cities. From my observation of Chinese media, it looks like the Chinese government is likely to reform disease control institutes like Chinese CDC, establish tighter biosafety regulations, and more strongly enforce laws protecting wild animals in the future. That would all be good. These issues are known to most Chinese American virologists because our attention has been largely drawn to this epidemic in the last few weeks. If you talk to any Chinese virologist here, they may tell you the same things. I may add, I think Chinese CDC is being wrongly targeted by the public, as ultimately it's some Chinese officials higher up than the head of the CDC who decide when to announce to the public that there are human-to-human transmissions. Because the whole country has literally been shut down by this epidemic, the public just needs some outlets to release their anger. It is probably less dangerous for the public in China to criticize CDC and scientists than to criticize the central Chinese government. There is so-called, I like this, there is so-called tossing the walk, the Chinese equivalent of passing the buck phenomenon on the Chinese internet for shifting blame from this epidemic from one entity to another. So that's all a very interesting perspective from a Chinese scientist.
And much of which we would not know, right? Much of this I have no clue about. I want to point out the Ralph Barrick experiment this individual is talking about. We talked about on TWiV. It's where they took spikes from SARS-like coronaviruses in bats and put them on SARS and showed they could infect human cells. And some people said you shouldn't do that, but that tells us the capacity of these spikes to infect human cells. It's important.
Yeah. I mean, as I read this, I think, okay, we need both the research and the public health component.
Mm-hmm.
Okay.
But of course.
And you got to do both of those.
But maybe not in the same institution.
Well, I mean, there could be a research branch and a public health branch in the same institution. I don't know.
Yeah, and I wouldn't say that our CDC does no research.
Yeah,
I mean,
sure.
No, they do.
So
yeah,
it's not it's not quite as split as it may have sounded.
Yeah. Anyway, thank you, Anonymous. We really appreciate it. We have a few more from individuals in and out of China, so that's cool. Can you take the next one, Kathy?
This is a different Anonymous.
It's a different Anonymous.
Okay. Hello, I've been listening to your podcast since the outbreak of the coronavirus, and I'm very glad I found you guys. It's reassuring listening from specialists in the field. I'm passionate about biology and everything related, although I never followed up with a career in this area. Just to say, I became a listener, and I do enjoy the chatting to lighten up a little. I currently reside in Singapore. The advice to health practitioners is to be on the lookout for people who traveled to China, Chinese nationals, or people who had contact with infected people. I have 3 young kids. One became ill in January, and a swab came back positive for influenza B. The doctor said that if they didn't have any fever, they could go back to school. So my questions are, 1, is it safe that the government is only testing the suspicious cases? 2, having been diagnosed with influenza B, does it mean they didn't contract another virus? 3, is it correct that after 24 hours of no fever, you are not contagious? In the Corona 2019 case, what is the process for being discharged?
Tough questions.
Yeah.
Mm-hmm.
So what does suspicious case mean? Fever? Someone with a fever? I don't know.
I don't know what triggers testing in this case.
I think fever is a big one, right?
Mm-hmm.
And
certainly if you had a CT scan and had a pneumonia, I would think trigger something. But that's pretty deep testing already.
So I would say that that's all you can do, because if you test, what's the alternative? If you test everyone, it's impossible. It overwhelms the system.
Right.
As it is, it's overwhelmed, right?
Mm-hmm.
So you have to have some triage criteria, and I think fever or pneumonia diagnosed by another method is a good way to do it. So from our perspective, that seems fine.
Right. In this case, we truncated some of this, and the child had fever and then fever and cough, but the cough was not severe. So, maybe more than just a fever or a very high fever, or it's hard. I think, well, Vincent, you pasted in the guidance for healthcare professionals. It's a U.S. document from the CDC that describes diagnostic testing and so forth, but I don't know if it describes, when should someone be tested.
No, it doesn't.
When not.
Unfortunately, they say contact your healthcare provider.
Exactly.
And they're really leaving it up. But, in the U.S., there aren't that many cases. So
right.
The real question is, in China, we just don't know what they're doing, unfortunately.
Yeah, I think probably what triggers the testing is, dude, I'm really sick.
Well, as you'll hear later, there are people in gated communities, and they're checked periodically for fever. And I would guess if you have a fever, they'll do a test, right? But if not, they're not going to test you.
I went to get my eye exam, and I do know what they do here. They asked, do I have a fever, and have I been to China, or have I been in contact with anybody who's recently been to China?
Right.
And I passed that bar, and then they let me in to get my eye exam.
Now, I would say if you're a family and one of them has fever, they will probably test all of you, right?
Yeah,
because you're all living in the same house, and
they should. They should.
And so, anyway, and you can imagine that extending to healthcare facilities. If a bunch of people are working together and one has a fever, they might test them all, and so forth.
Yeah.
The other questions, if you have flu B, does it mean they didn't contract another at the same time? No, you can get multiple, I think.
Yeah.
You know, but if you're getting well, then it probably would have just been one.
Correct.
All right. Then the other one is tough. Is it correct that after 24 hours of no fever, you're not contagious?
Oh.
So that's hard because for flu, that's what we do.
That's not true.
The problem is if you look for PCR positive material, you can find it, but it doesn't mean there's infectious virus, and it doesn't mean that you're transmissible anymore because you need a certain level of virus excretion to be transmissible. So the mere presence of a certain amount of RNA or virus even doesn't mean you're going to transmit it. And these guidelines have been developed based on, for flu at least, based on that kind of knowledge, that if you have no more fever, you're very unlikely to transmit. But I don't know what the situation is for coronavirus because we're very early on in this epidemic.
These are all numbers questions, right? You know, if you how much virus do you have? Because you know, with a lot of these viruses, I'm thinking of noroviruses, you can shed virus for a long time after your peak of illness, but you're going back to school. But you know, maybe you're not shedding as much virus.
That's right. That's the key. You do need to shed a certain amount to transmit. And so when you've got no fever, the titer has gone way down. And even though you may have virus, it may not be enough to transmit. Right. And in fact, the next one is about that. Can you take that, Dickson, from
Sure.
Liping?
Liping writes, I have been reading the latest Lancet papers on the Wuhan coronavirus. And I'd like to pick your brain on the following questions. 1. Patients still test positive after discharging and still shed virus. Does that indicate that this virus is similar to hepatitis B virus, such that we can be chronically infected with it? Whether or not it manifests itself depends on how strong our immune systems are. That's number 1. The number 2, I'm sorry, I don't know what the first word should be. They have found that the virus, they have found the virus in fecal material, and much like SARS in Hong Kong, there has been people infected with via the bathroom pipeline transmission route. Are there any known good practices to minimize the aerosol in the bathroom system? Well, there's some plumbing systems that don't do that. Thank you for taking the time. Fingers crossed that my questions get picked for the next podcast. Best regards, Liping.
So I don't know any evidence that this is becoming a chronic infection.
Right.
Right. So remember, we generally say a virus infection can be acute or chronic, and acute is resolved eventually, goes away. A chronic can last longer, even your lifetime. So we always thought that Ebola virus was an acute infection, right? You're infected and it goes. But then we found viral RNA, and in some cases infectious virus persisting in privileged areas like testis and the aqueous humor of the eye, where there's not much of an immune response. So, you know, our concept of acute infections are slowly evolving to, I don't know of any persistence of influenza viruses like that, but who knows? In this case, we don't know. We haven't looked at it long enough. And this saves patients still test positive after discharging. So I don't know if that's PCR positivity or infectious virus. And as we just said, may not be enough to transmit. However, that may go away in months. It may take actually months to clear it. In the case of Ebola, in some people, it takes months to clear that. So I think it's too early to tell. And I don't think it will be how long would a chronic infection be, right?
Yeah. It's, as you point out, this is becoming the difference between acute and chronic to some extent is becoming a gray area. Where do you draw the line? Yeah. Hepatitis B, it's been known for decades that people can carry a very active, but as often or sometimes asymptomatic hepatitis B infection for many, many years and infect a whole bunch of people. And that's known clinically, epidemiologically, et cetera. The viruses that we typically call acute, including Ebola, I think, or flu, don't don't behave that way. Now you may, carry under certain circumstances, carries some of these viruses around for longer than we previously thought, but it's nothing like hepatitis B. And I would expect that with the coronaviruses, it's going to look much more like an acute infection than like a chronic infection.
Now, as for the toilets, my understanding is that in some of the Hong Kong transmission was because there was a problem with the plumbing that was generating aerosol. And I think one of the issues was they didn't have water in there was no trap, the U thing that fills with water and allow. So those traps are there to prevent sewer gases from backing up, right? Yeah. And so if you have no water in the traps or no trap, someone else flushes their toilet and their excrement can aerosolize into your toilet.
I think there's a link later on because I opened a document later on in my list of things that's about, it's from the South China Morning Post, about a Hong Kong policy change on pipes. Public housing tenants were not allowed to modify bathroom pipes until August 2016. And then evidently after that, they were allowed to modify their bathroom pipes. And then it shows a bunch of pictures and some diagrams and things making you think that maybe letting people do their own plumbing is not a good idea.
Yeah, if you have a proper trap, you should not have contamination from other toilets. Because it is true, when you flush a toilet, there is an aerosol. Okay? So
this is very true.
But if you're not getting cross-contamination, so there's nothing you can do, I think, because if the plumbing in the whole building's modified, then you're in trouble. But this is not common, right? It happens in certain situations, and happened in Hong Kong during SARS. And I think there's been a recent incident with the new coronavirus, right, involving...
Right, yeah. So this is somebody on the third floor of a particular building, and she was linked to an earlier case of a man who lived 10 floors above her. And then they evacuated 30 households that had the same piping system with those two apartments.
Yeah, Okay, Gerald writes, hello, I appreciate your podcast. Thank you. I have two questions. One, what is it that makes the report by G et al. So unconvincing regarding snakes as a reservoir for the new coronavirus? It seems that their data did not carry much weight in your discussions. Okay, well, because the snake coronaviruses, their sequence is way different from the ones we're talking about on the phylogenetic analysis. And in fact, there's a new paper on bioRxiv, protein structure and sequence reanalysis of the 2019 corona genome does not indicate snakes as an intermediate host. So it was not only not a good bioinformatic analysis, but the snake coronas are too distant. That's why we didn't give it much weight. And now we know that this bat virus, well, we've known that for a while now, is the closest. Second question, if this outbreak were to gain traction in the U.S. the same way it has in China, do you feel we have the clinical resources to manage it? As a physician, I am concerned the U.S. public is getting a nerfed and edited version of the clinical issues and outlook. But I don't know what to say about that.
Yeah. What does he mean? What does he mean by nerfed?
Well, I don't know. It's hard to say, but my interpretation...
Look it up on Urban Dictionary.
Oh, nerfed is, I don't know. You're right. I don't know what it means.
Okay.
Maybe we're getting the wool pulled over our eyes about how serious this really is, and because I just don't know. Or maybe it's the opposite. My feeling is that we have the clinical resources to handle it, right?
That's my feeling as well.
Correct.
And so maybe he's saying, he or she is saying, everyone's going crazy over this, but why couldn't we handle it? So that's another interpretation.
Yeah, well...
Okay, nerfed means reconfigured. It comes from gaming.
Really?
So, yeah, I feel like there's a key phrase in his question is that if it were to gain traction in the USA the same way as it has in China. Yeah. My prediction would be that it wouldn't gain traction in the USA the same way it has in China, in particular, since we've been forewarned, because I think, we'd have it under control from the get-go, and we would therefore, keep it under control and have the resources to handle it. But, Could be wrong. Don't know.
Depends on the mechanism of transmission, too.
I keep thinking about the 2009 flu pandemic, right? That thing went global, lickety-split, didn't it?
Yeah.
Did, yeah. Mm-hmm. It was in a weird time of year, too. If you remember.
So I keep thinking back. Go ahead, Rich.
My sense is that this is not doing that. Is that, am I right?
Well,
or
it's very few. It's fewer than 1,000 cases outside of China, right?
Yeah.
That's amazing. I find that totally remarkable. And it has nothing to do with the virus. I think it's all about travel restrictions and control. And I mean, here in the U.S., if you have a fever and they take your history, as Kathy indicated, and you're from China, you came from China, they'll immediately quarantine you.
Right.
But I wonder if the same measures would have kept the 2009 flu geographically in check. I'm thinking no.
I don't know.
But I don't know.
We certainly didn't do that.
It didn't happen to my nephew, who moved back here one day after he returned from...
He didn't get sick, right?
Well, he didn't get sick or anything, but he could have been identified as a case potential, and he was not. And he went back to teaching.
I think there are far more flu infections. You know, here in the U.S., there are 15 million so far this year.
Right.
I don't think travel because it's not like there's zero travel. It's just from China.
No, that's right. So it all depends on the mechanism of transmission, though, right? Because if you look back to the introduction of West Nile virus in 1999, it's a virus that's vector borne. And once it's vector borne, you've got two things to control: people and it's a promiscuous virus that infects all kinds of animals. You're lost. And it just became endemic in the United States. And now it's just part of the regular scene in the summertime. Everybody accepts it.
It took us a long time to figure out. By then, it spread extensively.
Exactly right. But we knew it was vector-borne virus, though. We knew that.
Yeah.
We did. But we have trouble controlling vectors.
You know, that's an interesting case because we now know that every case of West Nile is mosquito to a person.
Absolutely.
Not person to person.
Well, organ transplants.
So that first introduction, presumably, it must have been a mosquito who came in.
Well,
because a person coming in would not have spread it to another person.
Remember, though, these little kids coming from Israel to visit the zoo, they had high viral loads and they didn't get sick. Little kids can tolerate a lot more virus than adults.
Mosquitoes are not supposed to pick it up from humans.
I know, not supposed to, but it's hard to imagine.
It's never zero. Is that what you're saying?
That's exactly what I'm saying.
Okay. All right. Okay. Sorry for the digression into flaviviruses.
Still, I mean,
actually, I'm not sorry.
If you don't know how this thing spreads, so now they've found it in fecal samples. What does this mean for the rest of the animal?
Where they got what? This coronavirus?
Yeah. And then how do they dispose of their feces in China? Certainly not as rigorous as we do.
I have no idea.
So that it has the potential for going back into nature. That's what I'm saying. Through discarding wastewater.
Yeah, but even though there is infectious virus in feces, again,
how long does it stay that way? Right?
How much is there? Is it
do we know?
Transmit?
Do we know?
I mean, it sounds like it has to be aerosolized to infect you, right? So, is there aerosolization in a sewage treatment plant?
Yes, there actually is.
Yeah.
For the aerobic digestion portion, there's certainly.
But this fecal transmission, first of all, it's a very small percentage of the overall transmission, and it's only under certain conditions where, like, the plumbing is defective. So I'm not too worried. Until we see it, I won't worry. But what do you want to do? There's nothing you can do about it. You can't piecemeal change the way they get rid of feces overnight.
No, you can't. That's very true.
Kathy, can you take the next one, please?
The next one is Cassidy.
Yeah.
Mm-hmm. Who writes, "Thank you so much for your common sense and logic in covering the 2019 new coronavirus outbreak in Wuhan. With the insanity of the 24-hour news cycle, I was so glad to find a podcast from researchers/practitioners, researchers in this field that was easy to listen to, full of great information, and provided a plethora of links to the actual research papers and articles covered. My mother is a doctor, my father is a science educator, and my personal background is in journalism and finance. But I'm currently an artist and run a small animal animal rescue farm for elderly dogs, all kinds of reptiles, and even a flock of poultry. All of this has taught me from an early age to believe strongly in going right to the source of the knowledge. You're all doing that, and it is awesome. Regarding spraying in China, here's one article I found that says in many places they're using bleach and other household disinfectants. They make a hydrogen peroxide product that was originally designed to clean up biological and chemical warfare agents, but is now being sent to hospitals in China to help disinfect and slow the spread of the virus. The product works on surfaces for up to 8 hours before degrading into non-potable water, so it lasts longer than a bleach and water spray. The article also does a great job of explaining why the widespread fogging and spraying isn't all that useful, wastes money and materials that could be focused elsewhere, and could give people a false sense of security. The crazy part is that they are not just spraying streets with it, they are also spraying hospital workers and patients with bleach water when they leave the hospital. And Cassidy provides a link. Indonesia also sprayed evacuated nationals with some sort of antiseptic as they disembarked from the plane before going into isolation on an island. And there's a link for that. Companies are also shipping robots to Wuhan for use inside hospitals to do disinfecting using ultraviolet light and disinfecting sprays. My local hospital actually has similar robots for sanitizing ER bays and patient rooms on most floors in between patients. And there's a link for that.
I didn't know that.
Twitter. I didn't know that either. Yeah.
No, that's new.
Wait a minute. We had a pick of the week some years ago, I think, with robots, disinfecting robots. Maybe it was my pick, so I remember.
Maybe.
They're pretty cool. They're pretty cool.
Okay. So
it's an R2-D2.
Yeah.
Yeah. Okay. There's one PS that I want to read. The Twitter is also found filled with examples of people who lied about travel history to avoid being put in isolation.
Yes.
Wonderful.
And yeah, one woman lied because she missed valuable time to be treated and died. And 44 people had to go into quarantine because of her. So this kind of plays up the point that Alan has made that, quarantine isn't always the best way to deal with things because you have unintended consequences. Keep up the great podcast, Cassidy.
This link she gives reference to the people being sprayed getting off of an airplane. Yeah, mm-hmm. Indonesia sprayed evacuated nationals with some sort of antiseptic is amazing. I just can't imagine getting off a plane and having all of these people dressed up like aliens spraying you as you get off the plane. It's crazy.
Yeah.
Rich, did you ever fly Qantas?
Do they do that?
They spray with insecticide. They fog the entire airplane before you get off. Damn right.
Huh.
They don't want anything flying out of there that doesn't belong in Australia.
Weird.
And yet they release rabbits. They release rabbits and then they release viruses to kill them.
And then they release another virus.
That was then. This is now.
2 weeks ago, this all comes up because 2 weeks ago we saw a picture of people spraying like a subway station or something like that and wondered what they were spraying and whether it did any good. And the answer is bleach, and in some cases, which gives me the willies, and in some cases, peroxide. And no, it doesn't do a whole lot of good.
Well, the latest fashion in China are white clothes. You get it? Because it's bleach.
Yeah.
They bleach.
Yeah.
Would you want to say something, Dickson?
Nothing.
Okay, let's, Rich, can you take Brendan's?
Brendan writes, and I want to note that this came in on the 10th of February, so it's a little bit dated, but it's a great personal perspective. Hi, Twivers. Wanted to send you an update from Chengdu, as well as ask about possible spread of this virus between mammal species. Sorry in advance for the lengthy email. First off, the weather's very pleasant here with temperatures in the mid to high teens Celsius and mostly sunny skies. Life here has continued to be under mostly self-imposed, mostly quarantine. All non-essential public spaces remain closed. That's pretty much everything except hospitals, convenience stores, and supermarkets. I heard a joke in Chinese which paraphrases to, Your Chinese New Year vacation is almost over. Alert, congratulations, your vacation has been extended. Given the coincidence of this outbreak with the holidays, that's really what it feels like. For those who haven't spent time here, note that Chinese residences are primarily clustered in gated communities. The social, regardless of income level. The social distancing measures have become much more organized lately, with each housing complex requiring residents to get a temporary ID card for entry and exit. Everyone going in and out gets a temperature check, but those who don't live there go through a longer health check and registration process before being allowed in. Also, yesterday on February 9th, the police came and did a separate door-by-door registration. This was ostensibly to do an update census of where everyone is and aid with tracking contacts of infected individuals. For the moment, we still have limited freedom of movement around the city. Public transit's still running, but I'm braced for that to change at any time. And remember, this was February 10th. I don't know what's happened in the meantime. I can't tell anything about the efficacy of these measures, though. As of around February 5th, Chengdu has confirmed local human-to-human transmission with 120 cases, and the growth in cases per day is around 5%. That's about the same as Wuhan, although the magnitude is much less. If you look at the curves on the Hopkins website, it does look like the case growth is slowing, though. This is all with the caveat that the confirmed case metric is problematic. As you all have noted, the figure is certainly underreported. Also, there may be some bias in testing and or reporting between areas. I could imagine relatively affluent cities have more testing and overreport compared to lower income areas, for example. Chengdu might be really better off than other parts of China, though, as the Sichuan province governor Yan Li has public health background. He was on the SARS expert panel in 2003, after which he headed the Chinese FDA before becoming governor. While the official response has been problematic, early January in Wuhan, for example, I want to echo the shout out on TWiV to the public health community here. For now, it seems that they are being given a lot of leeway to do their best. Having regularly listened to TWiV for a while, I'm personally not too afraid of this virus. The thing I worry about is the public reaction and the impact on the economy. Businesses were supposed to open today on February 10th, but that's been pushed back. Based on how the political system works here, I foresee that remaining status quo. This is because regardless of the efficacy of control measures, the political risk of letting businesses operate and then seeing an uptake in cases is too high for local authorities. However, as Vincent noted in his virology course video the other day, if the ratio of confirmed to total cases is 10%, then maybe the ultimate control measure will end up being immunity. Personally, I'm rooting for some of the antivirals being trialed in Wuhan to show efficacy for reducing severe outcomes. If that happens, then maybe this starts to look more like seasonal flu in terms of severity, and people can get on with life. Speaking of getting on with life, I was out for a walk the other day and I saw a guy struggling. This is great. I saw a guy struggling to put a doggy surgical mask on his husky. At first, I laughed to myself. Then I thought, well, there must be a non-zero chance that this could infect dogs, higher than snakes at any rate. Have dogs been investigated for other human pathogenic coronaviruses before? Anyway, thanks for all you do. I'm almost caught up to the 2014 Ebola outbreak in the TWiV back catalog, so that will be interesting to listen to in parallel with this. All the best, Brendan.
Oof.
Nice email, nice perspective from on the ground.
It almost sounds like a description of what was going on in London during the Second World War with Edward R. Murrow sitting on the roof.
Yeah.
Well, there are dog coronaviruses, but they are not human coronaviruses.
Right. The highly infectious intestinal infection in dogs, especially puppies.
Yeah. So I don't think he had to put a mask on his husky, but fine.
No.
If you want to do that, that's okay. Well, thanks, Brendan. We appreciate the updates. I wonder what's happening now.
Yeah.
Right.
Dickson.
Yes.
Bonnie is next.
Bonnie writes, Dear Twivsters, thank you for the excellent science-based info on the COVID-19 situation. I am an infectious disease doctor and would like to point out an important factor in diagnosing this infection, and in parens, all respiratory pathogens that use nucleic acid probes for testing. This is what this
Specimen collection.
Oh, I'm sorry.
That's important. That's
Absolutely.
what she wants to point out.
No. Okay, fine. Okay. Specimen collection. This is of particular concern now that China has transitioned to a much more problematic clinical diagnosis based more on clinical symptoms than PCR testing. While it may seem trivial how a healthcare worker jams a swab into someone's throat or nose, technique is important. If anterior nares, front of nose, swabs or side of mouth swabs are done rather than the true posterior nasopharynx or oropharyngeal swabs, the sensitivity drops dramatically. We have shown this to be true repeatedly with diagnosis of influenza and other respiratory pathogens, and I am disappointed that it has not been mentioned more in discussion about the PCR tests. Missing the diagnosis and stories about people who are positive, then negative, then positive again. We often see patients admitted from the ER with the perhaps carelessly collected flu test who miraculously are flu positive by the time they are admitted. Like so many cultures and diagnostics in infectious diseases, specimen collection and handling is critical. I warn my patients about the unpleasantness of a nasopharyngeal swab and, jokingly, tell them if it doesn't cause a wincy face reaction, we haven't collected specimens from where the viruses reside. Thanks for helping to get the word out. P.S. I am sorry Vincent feels MDs are not scientists. While most of us are not researchers, to be sure, this MD certainly knows the mechanism of action of acyclovir and many other mechanisms for the hundreds of antimicrobials, etc. I prescribe. I am sorry if so many of you med students have already lost the love of science that inspires many of us to choose our profession. It's the science and the cool case wow moments that get us through the day many times. Thanks for promoting the science in all things microbe.
Yeah, it's very important, this specimen collection. It's great. Thank you. Yeah. I'm sorry, it's not an insult, but it's true. Scientists do research. You don't want doctors to do research on patients. I wanted to also point out from Brendan's email that in my course now, my virology course, I've gotten into this habit before every lecture. I give a little update from the Hopkins website and make a few comments because this is very cool that I should say it's very instructive. It's very instructive.
Sure, absolutely.
Whenever possible, I incorporate some of the papers that are coming out into the lecture. And, this ribosome binding domain business, we've already passed entry, but I'm trying to figure out how to get it in somewhere else because it's very exciting. And in emerging infections, this will be great.
You didn't mean that as a pun, I hope.
What's that?
It had already passed entry.
and once again, you said ribosome binding domains. Exactly. You meant receptor binding domains.
What is this story?
Did you do a translation lecture today or
no,
something like that?
No.
As Kathy and I have discussed just today, I'm not doing a translation lecture anymore.
It was lost in
done one since 2017.
Lost in translation.
Receptor binding domain. Judy writes, I live in Singapore, and as of this moment, there are 43 cases, of which 6 ended up in the ICU. Given the government here is highly functional and transparent, I trust these numbers more than any coming out of, anywhere, including China. This implies a 14% rate of cases becoming severe, though fortunately there have not been any deaths. Also, several cases have mild symptoms for a week before being severe enough to be sent to the hospital for screening. In all the stats I've seen about this virus, none mentions what percentage of cases go on to require critical care. This would make a big difference in death rates, especially compared to regular flu. If, in the case of Wuhan, the health system is in crisis, that would mean a higher death rate. If, as in Singapore, that could mean a higher survival rate due to the availability of supportive care, right? This makes the death rate highly variable depending on how widespread the virus becomes and consequently, and more importantly, how overwhelmed the local healthcare resources become. It would be interesting to see if this statistic exists outside of Singapore. I tried to Google, and all I get are numbers of deaths and infections. No rates of cases requiring ICU or becoming serious, however that is defined. I don't know, maybe that paper we did.
I think that paper has some of those data in it.
Yeah, it's the China CDC paper we talked about earlier. Concerning how many have had mild symptoms that only become severe after a week, in Singapore, there are several cases where people went to an urgent care clinic with mild symptoms 2 or 3 times before becoming sick enough to be sent for coronavirus screening. These folks were not profiled as being susceptible to the viruses. They had no recent travel history or contact with anyone from China, so were not sent for screening immediately. I haven't seen many stats requiring onset of initial symptoms to being severe enough to be diagnosed. There are maybe thousands of folks infected with coronavirus who have a mild cough and fever, think nothing of it. Does that mean in 7 to 10 days we'll see a spike in cases that become severe enough? Folks will seek treatment at the hospital. Does this explain the apparent low rates of transmission outside of China? Is it just because there hasn't been enough time for the severe cases to manifest and be found? I don't think so. By now this would have all shown itself, but we haven't seen that happening. I'm sure WHO is all over this, but I'm wondering why there's so few articles asking these questions, or maybe I'm not seeing them as most of what I'm looking at are mainstream publications. Would love your expert commentary. P.S. Being in Asia closer to China, to the China pandemic, folks here are panicking a bit, so it's hard to escape the news.
It's actually hard to escape the news here. It's like 2 full pages in the New York Times every day, minimum.
Right. Well, it's in the Financial Times also.
So I don't know how anecdotal these are, Judy, because you're in Singapore where that many cases compared to China. And so I don't know that for every case you have to come back multiple times before you get serious. As you know, the incubation period is an average of 5 days. So, she suggested that this might explode at some point, but it hasn't. I think this email is.
I'm looking back at the Hopkins site, and on January 19th, this is on the leading edge of the daily cases curve. There were 282 new cases, and that sort of epidemic curve peaked on February 4th. So that's 2 weeks later. And that peaked at 4,000 new cases, going from 200 a day or 50 to 200 a day, depending on which day you look at, up to 4,000 cases in a matter of 2 weeks. And I think we would have seen that somewhere else by now.
Yes, I agree.
If it was going to take off somewhere else. I say all of this stuff, hesitatingly, because you're trying to, I'm not an epidemiologist, okay? And this is a new deal. I don't know what's going to happen, but it looks to me like if it were going to seed itself in the same kind of way outside of China, there would have been time enough for that to happen by now. We'll see.
So this next one, I think we've answered most of this. So incubation period or not. Wearing a mask. All right. So what do we think about the travel restrictions? Well, they seem to be working, but initially I was skeptical. But good thing I'm not in charge.
Well, travel restrictions, unless it means restricting people on a cruise ship.
Correct.
Yeah. So we said when that happened weeks ago that it was a bad idea.
Very bad idea.
Let them go home and quarantine them.
Very bad idea.
And now we know it's a bad idea.
That Japanese doctor that went on board that ship came off with his eyes wide open. He didn't want to go back.
You can't do that because the air is not going to be cleansed. Exactly. It just circulates among everything. They should have sent them home and quarantined them. But we did say that 2 weeks ago. Wearing a mask. Yeah, this is an aerosol spread infection. Yeah, for sure.
Wow.
Wearing a mask is fine. It will prevent spreading of aerosols. So you can do that. Kathy, can you take the next one?
Anthony writes, bats shedding viruses. The Central Park Zoo has bats and provides a link. I don't know how large the enclosure is. The guano certainly can be collected without disturbing the bats. The bat's resting temperature can be determined, albeit not precisely, non-invasively. I'd hope that the guano is monitored for viruses and the temperature is checked, as this information could be valuable. I speculate that pregnant and nursing bats shed more virus.
So this was because we were wondering whether, resting bats shed a different amount of virus from excited and flying bats. And so he's suggesting we go to Central Park. Mm-hmm. However, in Washington, someone told me that the farmers in China harvest bat guano from caves for fertilizer. And these are intestinal infections. So I wonder how much virus is being transported around that. Is that true that they use it for fertilizer, Dickson?
They'll use anything organic, but yeah, they do.
They go in the caves of the bats and get it?
I presume so. I don't know this firsthand, but I've heard rumors to that.
So if they then go to market, they could be infected and transmit it, right?
Where do they get those bats that they do bring to market? The actual bats themselves, right?
All right. So Cam, we did.
Yes.
Rich, can you take Mario?
Mario.
So Kathy, after this, we'll let you pick and then leave, okay?
Okay, sounds good.
Hello, it's -9C here in Montreal, and we're buried under about 15 centimeters of snow. I am a longtime fan of your show, and I'm really happy you folks are doing this series on 2019 nCoV, which is now SARS-CoV-2. My question is on the impact of poor data quality on decision-making during times of major outbreaks and responses. I have noticed a very heavy reliance on official Chinese government numbers for predicting mortality rates. In the last couple of episodes, it was stated a few times that the upper limit on the mortality rates are around 2%, and that the number of infections seem to be contained to China. And these 2 points have been repeated in the press often. Do we have a bias for over-relying on the official Chinese government data simply because it's the only quantifiable data we have? Yeah.
What else do you want to
Because there seems to be a very significant amount of contradictory anecdotal data leaking out as well. And while it is poor in quality, there is much more of it, and much seems to contradict the official numbers. You mentioned in your previous episode that there is likely an undercounting, both due to limited testing resources and also because the data is curated centrally. But the team, like the media, always seems to circle back to the Chinese data anyway. My question is how, in such a chaotic situation, do experts in outbreak response factor anecdotal data sources into their decision-making, particularly when they contradict the official numbers? It may seem like a challenge both from a statistical perspective as well as a diplomatic one. The question was asked on the show, why haven't we seen more cases internationally? I wonder if in the countries we owe so, I wonder if in the countries that owe so much of their economies to tourism, such as Nepal, Thailand, Vietnam, Laos, whether they are less vigilant in reporting the virus out of fear of sabotaging their economies. Thanks. Well, my understanding is that there is there's a central Chinese government reporting system where cases are reported. And, I think that that's going to be the most reliable source of data. That's why I go around to it. I wouldn't pay a whole lot of attention to, I don't know what anecdotal sources he's talking about, but I would go for, the official sources. That's the really the best you can do. Whether other countries might be underreporting to keep themselves to protect their tourism, I doubt it.
Problem is anecdotal is anecdotal.
Yeah,
and I don't believe in social media trends. Yeah. I just think in the end we have to depend on the Chinese source, and when it's over, we'll get serological surveys, and we can look back and see how off it was. But it doesn't make any sense to see on Twitter someone says there are more cases here, and we can't do it. We can't figure out if it's true or not. All right, Kathy has to leave us, so let's get her pick of the week.
Okay, I picked something that's going to be in a way related to Rich's pick, and it was totally independent. But this one is called the Deep Sea. And when you go to this site, you can start scrolling down and see different organisms at different depths of the ocean.
Oh, neat.
And you keep scrolling and you keep scrolling, and then the organisms kind of thin out, and you keep scrolling, and you look over at the scroll bar, and you have a long way to go. And when you get down, I mean, then, yeah, they really do kind of, fizzle out. There's not as many. But I was down at 8375, I think it was, and there was some organism. And then I went a little bit farther, and it says, you have now scrolled the height of Mount Everest.
Right.
And I don't think there were any things there beyond that depth. But I did this first weeks ago when I first got it. But it's really fascinating.
This is awesome.
This is a great
awesome.
site.
But if you get to the bottom, there are stuff there.
There are stuff.
There are stuff.
I was just astonished by some of this stuff. That an elephant seal can dive to 2,400 meters.
Yes.
Yeah,
Okay.
cool.
I mean, the pressures at those depths are phenomenal.
What about sperm whales? They go deep too.
But not
I have to look them up. And when you start getting down here, the comments on this are very interesting.
the sperm whales go 935 meters, but there are things that go deeper.
There are. Yeah.
Whoa.
You're right. You're right.
Like anglerfish. I think it's cool. And then, of course, the sub went all the way to the bottom.
That's true.
Yeah.
It's true.
This is great. This is really cool.
Yeah. Yeah.
It's very nice. Thank you.
But the first time around, I didn't pass that. I didn't see the thing about
Pretty funny.
the depth of Mount Everest. It's just amazing.
Yeah.
I kind of started, and I was kind of trying to take it all in, and then I saw where I was and how far I had to go. Wow.
Yeah, I'm going to show this one to my grandkids today. This is great. This is really good.
All right. Thank you, Kathy.
Yep.
Okay. Thanks. This is a lot
Thanks,
of fun.
Kathy.
Yeah. Bye-bye.
Okay.
See you next time.
Okay.
See ya.
Bye.
Anton writes, now there's a significant number, 463 cases of corona outside of China. The death rate in China is 2.38%. The death rate outside is 0.4%. Why? Are we helping China enough? I think that China is overwhelmed.
Yeah.
And the cases outside are spread out and they're easily cared for, right? What do you think?
I would agree.
That's the simplest explanation, consistent with
agree.
Everything we know. Yeah.
All right, let me take the next one from Rarity. Hey, Rich, you okay with going on some more?
Oh yeah.
You okay, Dickson?
I'm retired.
Yeah, I might just
got
have
all
to come
kinds of
back
stuff.
here and find out where we left off.
Ruarid. I started listening to your podcast when this latest outbreak started. Thought you might be interested in an update about what life is in an average Chinese city. I live in Suzhou in Jiangsu province, a second-tier city, urban population 2 to 3 million, metropolitan population 10 million. There have been 80 cases here. City for the last week has implemented a curfew system where only one person per household can go out once every 2 days to buy essentials. In addition, many roads have been blocked, funneling traffic and pedestrians to checkpoints where you must give your ID, submit to a temperature check. All restaurants and public places are closed apart from McDonald's, Burger King's, and KFC. Leaving the house,
Nothing lives inside of those restaurants.
You must wear a face mask, and you are temp checked if you go into a supermarket. My uni classes will be online for at least 10 weeks, and it seems the primary and secondary schools are doing the same. Although there are a few cars and people about, mostly the streets are quiet and deserted.
Good.
I've had photos from around the city. Hope you're well. Yeah, so these are interesting photos. There are not many people on the streets.
Goodness.
Vincent sends us a nice website that analyzes the phylogenetics of the coronavirus outbreak. This is nextstrain.org/NCOV. And it's basically the genetic epidemiology. All the sequences that they have are arranged by date on a phylogenetic tree. You have global tracking. You have protein diversity. Really nice site.
Yeah, amazing.
You can choose how you want it displayed and so forth. So
Cool.
It would take someone named Vincent to send this. Vincent is in Germany.
Yes.
Dickson, Matthew
writes, Dear good people at TWiV, grateful for your intelligent and informed discussion about COVID-19 SARS-CoV-2. Would like to hear your thoughts about the nationwide spraying of bleach and other disinfectants across China. Apparently, this is not nearly effective as actually wiping down surfaces in the places the virus is most likely to have settled on. And what about the health dangers of inhaling bleach? Even if diluted, surely not as bad as COVID-19, but can't help but having a major impact on dumping that amount of chlorine into the streets, waterways, and nasal passages of an entire country. Thank you again, Matthew. As you can tell, I'm a little congested, but that's okay.
Yeah, we talked about it in that good email we got up above. Yeah, absolutely. This is not great.
Look, you'll recall that during the original outbreak of, I keep going back to the West Nile virus, they started to spray malathion from helicopters all over the Manhattan Island. That was just crazy. It's a panic attack from the lack of knowledge. Basically, that's what that results from.
Rich, can you take Gail?
Gail writes, biology is not my field, but upon reading about testing issues, perhaps this event could be informative. Evidently, the infection was spread within an apartment building in Hong Kong by toilet wastewater vent. Someone thought it needed improvement and did a bad job. Perhaps the best way to test for infection is by using feces. It's a lot easier than to collect from a patient than trying to sample lung mucus. Well, interesting point. I don't.
So you don't actually sample lung mucus. You do a nasopharyngeal swab.
Yeah,
which I think is easier than getting a fecal sample from people.
Yeah, right on the spot. Yeah,
and the toilet spread. The feces is not every transmission. It's only a small part. So you got to do the nasopharyngeal swab. Yeah,
and you got to do it correctly, as we've already learned.
Yep. Sophia writes, "Dear viral experts, thank you for helping me keep my sanity these past days. I will be eternally grateful for your help. I have a couple of picks. I'll read them now in the interest of time. One, free WHO online courses, not as informative as TWiV, but quite useful otherwise. openwho.org/courses, and to the WHO Facebook page. They have useful infographics and had a nice piece about receiving packages from China. No virus threat. Quite early in the epidemic, we said the same thing. They also had a Q&A on traveling, which was quite informative. Talked about flying cruise ships, etc. Having said that, I was quite confused by some of the statement, and I have some questions below. One, the virus survives for about 30 minutes on surfaces. No one knows about this particular virus, but SARS and MERS last longer. So that's not right.
Right.
Especially
again, we did this and came up with a sort of a number of 5 days.
Yep.
Last time.
2, there's no such thing as an asymptomatic person. There are always symptoms. Even if mild, they have subclinical symptoms. A person transmits as long as they have symptoms. Well, I'm confused now. What do you guys think? Wrong. There is an asymptomatic person. There may be signs. That's different from symptoms. A sign is not something that you can tell someone, because it's like taking your blood and looking for cytokines or something like that. So there are asymptomatic persons. Subclinical is another way of saying that. But signs are a different story, as we said earlier. And they say a person transmits as long as they have symptoms. It depends on the infection. So influenza, you transmit about a day before you have symptoms.
Yeah.
And other viruses you do not transmit.
In this particular case, we don't really know yet. I don't think there was a report that asymptomatic people could transmit, but that report had flaws. We don't really know.
We'll find out. But Ralph Barrick said for SARS, you mostly transmit at the peak of clinical symptoms.
Right.
That's the transmission. So this is wrong from WHO.
Ooh. Thanks
for all you do. The latest episodes are the epitome of public service. All the best from Greece. 6 degrees C. All right. Now she has a few more questions. WHO is saying they're trying to contain the virus in its source. I can't remember if they did that in 2009, but is this even realistic? What's wrong with declaring this another respiratory mild infection might get every year? With a fatality rate this low, why treat it like Ebola? I don't know what it means to contain it in its source. That would be China.
Yeah, I don't know what that means either.
So this could establish itself in humans. really depends on whether it's all gone after the winter season is over or whether it is transmitted over season like many other viruses do. There's no way of knowing, and we just have to wait to find out.
Indeed.
And, I mean, the fatality rate seems low. Certainly it's lower than something like 60% like Ebola. But if it turned out to be, in reality, just to pick a number out of all the numbers out there, 1%, okay? If you infect everybody with it and there's a 1% case fatality rate, that's mind-boggling.
That's a lot.
That's a lot of people.
That's about Rich, you just gave the statistic for malaria. Okay. It's got a 1% mortality rate, but there's millions and millions of people infected.
Yeah. So that's not trivial.
No, it's not trivial. Absolutely correct.
Okay, let's do a few more here. We'll
wait a minute, she's not done yet.
Oh, she's not done. Sorry. I'm glad you would go to Wuhan. How come everyone else is canceling events and conferences in the nearby area? Shouldn't WHO make a statement about safety in conferences? Yeah, they are canceling conferences, not just in China, but
everywhere.
Everywhere.
That's
especially, right. I know someone who was going to a conference in Malaysia that was canceled. Yeah, they don't want people gathering.
One canceled in Nottingham, England.
Yeah,
individually, personally, I would take the risk to go to Wuhan and do that and come back. I might suffer some consequences when I returned, and people, wanted to keep an eye on me. But I can understand that, having an event that brings in hundreds of people and then sends them all home would be discouraged. In particular, bringing them in and sticking them in one place for a period of time,
Yeah.
How do you like the new name? Well, we talked about that. She wants to call it Wuhan virus. No, I don't think so.
That's not going to work.
They would not like to have it reminded forever that it started there. So
yeah, that's right.
It's not good.
That's right.
Who's next? Did you read one recently, Rich?
I don't know.
We'll take it anyway.
Dickson is next, yeah.
I can do it next.
It's long, but...
Sabrina writes, "Hello, TWiV team. I love your podcast, and while I have wanted to write many times, I felt like I had some possibly useful commentary in regards to some discussion at the end of episode 586. You were discussing flu monitoring and reporting in the U.S. tangentially to an email asking if cases of nCoV were possibly/probably being missed in China. I'm a clinical laboratory scientist at a small community hospital in Florida. We run our flu tests via PCR assay and report for flu A or flu B. We do not subtype. As I am the head of my microbiology serology department, I have to prepare weekly reports for our infection control department, which in turn is provided to the DOH, Department of Health. My data is pretty basic, though. Simply numbers of flu tests performed for the week and how many positives for A and B. Patient samples are kept for 7 days and then discarded. And I have never once been asked to provide samples to the DOH or to any other agency for subtyping. Again, we are a small facility, so this may be part of the reason. We did have a fatality this season that appeared to be possibly flu-related. I asked my infection control department how and if we brought that to the attention of the DOH for flu monitoring purposes, and I wasn't given a clear answer on how that was reported or followed through on. All of this is to simply say that I can definitely imagine that the coronavirus numbers are imperfect because I can see a lot of areas where the way we report flu statistics is open to interpretation and individual hospitals' understanding of how and what to report. In response to coronavirus testing for patients, I obviously don't know how the system works in China, but here at the hospital lab can't just start running a new test out of the blue. Aside from the test having to already exist and to be manufactured, produced in substantial volume to meet demand, the test has to be approved by the board of directors of the hospital, validated in the laboratory by running predetermined numbers of known positive and negative control tests. SOPs have to be written and supplemented, and the staff has to be trained on the new assay. We currently had not had any patients tested for the 2019 coronavirus, but we have had a few physicians ask how to go about ordering the test if they wanted it. At this time, a sample would have to go to a state reference lab for testing. I'm trying to picture how this would play out in a facility similar to mine in size and scope in China during an outbreak, and it is not hard to understand how the system could become overwhelmed quite easily. As we are in flu season, our little lab is already stretched thin and very busy this time of year. We would be very hard-pressed to rapidly onboard a new test and deal with the added volume that would result from an outbreak such as the one currently going on in China. So my heart definitely goes out to the boots on the ground at the clinical laboratories in China right now and to the people trying very hard to gather correct and useful data that will paint a workable picture of what is happening with this virus. Because I know that when we are looking for the overall numbers and the cumulative data sets, it is sometimes easy to forget all the moving pieces that contribute to bringing all of that information together. Thank you for letting me ramble and for your brilliant approach to communicating science. Warmest regards, Sabrina.
Great points.
Yeah, great. That's a wonderful perspective.
Yeah, absolutely.
So this idea about having to validate a test, it's true, because I contacted someone here,
of course.
And he said, it takes months.
At least.
You have to, first the company has to develop it, they have to test it, then we have to validate it, then the Department of Health has to approve that we use it.
Correct. So what does happen in this country? What does it do? Just get sent to the CDC?
Yes. So they will do the testing. They'll do the PCR.
Yeah.
Although they have published the protocol on their website, so I don't know what that means, but I don't think you could use it for official. A hospital would not do that. It has to be
that's right.
validated.
That's right.
Rich, Poppy.
Dear respected TWiV team, my first time writing, long-time listener and learner. I used to listen to TWiP, TWiV, and TWIM in the garden as I pulled weeds and trimmed shrubs in the summer. Now I listen to it while I'm cooking in the winter. Cool. My question is, if the coronavirus makes us humans this sick and dying, does it also make bats sick and dying? Would one find a ton of dead bats all over the place? And could you address the changing of method of counting the sick, which made the number of sick jump dramatically the night of February 12th? I just read the news, and you know how it is. Don't know how one can trust anymore. Thank you especially to Vincent, who got these podcasts to educate the public like me. Poppy. Okay, so first of all, it's not exactly the same as the bat coronaviruses. Second, it is the nature of these things that a virus can find an equilibrium in an animal host where it doesn't make them real sick and then spills over into another host. And in that unfamiliar environment, if it gets a toehold, things go wonky and it can make you very sick. So that's a pretty common circumstance and characteristic of a spillover. Asymptomatic or very mild symptoms in one animal spills over into another animal and causes a problem. And we commented on this method of counting the sick earlier and about how we didn't really approve, and it turned out that others didn't either, and they backed off on that.
Yeah, the bats have lots of these coronaviruses, and they seem to be fine. But whether this particular human one would make him sick, I don't know. I would guess not, but no one's going to find out, I guess.
Right.
Jen writes, thanks for your very informative podcast. I recently discovered it due to COVID-19. Enjoy it immensely. Do we yet know if SARS-CoV-2 is self-limiting? A very concerning rumor is that a Chinese health official was sacked for stating that it is not self-limiting, and provides a link for that. Of course, it's self-limiting. Eventually, you either die, and not many people are dying, or you build immunity, and it's resolved.
Right.
And it does. I don't think it's causing a persistent infection. Okay, that's what the rest of the email is about. Speaking of the lack of education, what's good intro to virology textbook for someone unable to pursue further higher education who has undergraduate degree in biochem, interest in spare time? Any open courseware courses, local conditions, Copenhagen, bright, sunny, 5C. It's been 5C all along, but it's always gray and overcast. Thank you for your work. Go to virology.ws/course. I put my virus lectures there. I teach a course here. They're up on the website for free. And you can check that out. And for a virology text, well, I have written one with some co-authors. It's
2 volumes.
2 volumes. Maybe it's a bit complicated, but most of it is covered in the lectures. So why don't you start there?
Yeah, do the lectures.
Good idea.
What do you think? All right, we're going to skip the next one because Kathy wants to comment on it. Let's go to Stuart.
Stuart writes, "Hi, guys." That's a bad start. "Hi, everybody. Great pod as ever this week. Thanks for TWiV and all the other TWiVs. Question on potentially slowing the spread. You told us the entry point for the virus is the human membrane-associated ACE2 receptor. We have dozens of ACE inhibitor drugs available, initially developed to treat high blood pressure. Could we not provide ACE inhibitors to people exposed to the 2019 nCoV carriers and see if that reduces transmission? Is it probably no use for treatment of infected subjects, but by blocking ACE2, could it reduce infection rate in newly exposed people?" From sunny Raleigh, North Carolina, 57 degrees Fahrenheit, mostly sunny.
Nope, can't do it. It's been tried with SARS, coronavirus. ACE2 inhibitors bind at a separate site on ACE2 from where the virus binds.
Thank you.
It was actually someone in my class asked me that during lecture, and I said I have to look it up.
Smart.
While I was lecturing, she looked it up, and then at a pause, she told me the answer. Isn't that great?
I love it. That's great. I love that too. That's wonderful.
Glenn, I'm an expat New Yorker. Started following you in the good old swine flu days. These TWiV episodes are like pure uncut blank. Enter your chemical vice to an addict. Thank you very much. My question is, having read about corona shedding in feces and about the SARS incident where a cluster occurred in an apartment complex, which we have mentioned already, how likely or plausible is the spread in these markets by this means, where all manner of critters leave their mark? So in other words, are the animals shedding it in their feces?
It's a good question.
It's a good question. We don't really know how many of these animals are shedding. The bats are certainly shedding these. It's an intestinal infection in bats, right? However,
you got me.
Yeah, it's an intestinal infection in bats, and so it's in the guano. Right. Whether it's in the excrement of other animals, we do not know. However, in the meat markets, eventually the animals are slaughtered, and that really spreads virus everywhere as they're cutting them open and their organs and so forth. So yeah. I don't know if you would particularly need feces. Who's next? Rich, Stacy.
Yeah, and I am going to have to. I just got a note from my wife who was elsewhere that I'm going to need to leave pretty soon.
All right.
And go pick up my grandson at school and show him Kathy's pick.
Okay, so can you do one more?
I can do, yeah, sure.
We'll do picks.
Yeah.
Okay.
And then do picks. Yeah, that's fine. Stacy writes, "Hi, TWiV team. Thank you so much for answering my questions. That was incredibly exciting for me. I have a 2009 study for you to read about long-term lung damage caused by SARS and 2 more questions." She gives a link to the study. My questions are, is the new COVID-19 virus showing the same level of lung damage in patients who have recovered? 2, does smoking cigarettes increase the severity of coronaviruses in general? I don't know the answer to number 1 and I don't know that is known.
No, it's not at this point.
And smoking cigarettes, I can't imagine that it wouldn't increase the severity of coronaviruses in general.
So that paper we discussed where the men, most of the men in China smoke 52%, right. That could be it, yeah.
My concern is that China is going to have an enormous number of citizens with an impaired lung capacity, which will put further pressure on its healthcare system over the years. I'm firmly on the side of keeping conversations as part of each podcast. As a non-scientist, it is a tremendous pleasure to hear the passion that drives your curiosity, and I laugh out loud as often as I gasp in wonder at what you're discovering. Thank you for trusting that listeners will understand and care. Stacy, well, that's very nice. I appreciate that. I think about what we do here And what I hope, as much as anything else, is that what people hear is scientists exercising critical thinking. Okay? And saying stuff like, "Oh, I was wrong about that," or "I don't know."
That's
or,
my
"Let's see how
not yours.
How do we figure that out? You know, because I think one of the most important things we can possibly contribute, aside from the fact that scientists are real people, is the importance of critical thinking and how it's done. So,
yep.
Good.
By the way, this paper is about long-term sequelae of SARS, and impairment of lung diffusing capacity persisted in 24% of SARS survivors.
Wow.
Their exercise capacity and health were markedly lower than the general population 1 year after illness. So if you have serious lung disease, and especially if you are older, I would guess, and have other issues
and stuff, that's right.
It's a problem. And so your point is really good. This could have, however, 80% of these infections are mild.
Right.
So. Right. And even, I'm doing the math here, 75,000 cases, right?
Yes.
How many people are in China? 1 billion?
Yes.
A lot.
It's a very...
Many.
It's I don't want to, if you're one of the people affected, obviously it's 100% for you. Okay.
Yeah.
But still, it's compared to flu, it's not that many people.
No, it's not.
Yeah.
Okay. We have lots more email from you. We appreciate it. We'll get to it next time. Many of them are the kind of question that don't go out of date, fundamental stuff. So we'll get back to those. But now let's do some picks so that Rich can go. I think this is now the longest TWiV ever at 2 hours and 20 minutes. Yeah.
I almost got here on time.
Yeah, you did. I appreciate your sticking around. Really.
I love doing this.
You guys don't get paid and you just want to teach.
I don't get paid.
Some people yell, and these are not emails we read, but some people hate that we chat all the time.
You know what?
I think that it's a part of the scientific conversation is that we have chats.
Exactly.
Rich, what do you have for us this week?
So I have a pick. This was just kind of lying in my TWiV picks box. That was actually this is from the Guinness Book of World Records, published in March of 2018, that reports the world's oldest message in a bottle confirmed 132 years after being thrown overboard. Now, that may not sound like a science pick, but the deal is that there was a drift bottle experiment conducted by what was the German Naval Observatory between 1864 and 1933, when thousands of bottles were thrown into the oceans from German ships, each containing a form that the captain would write the date it was jettisoned, the exact coordinates and time, the name of the ship, the home port, and the travel route, and on the back ask the finder to say where it was found and return it. Okay? So this is, the old time version of trying to figure out what the currents are doing. This is fantastic. And somebody found one of these bottles on a beach in Australia.
Wow.
132 years after it was thrown overboard.
I'll be darned.
It had traveled a mere 950 kilometers, apparently. And the suspicion is that it probably only drifted for about 12 months and then got buried in the sand for 132 years. And then somebody dug it up. But the cool thing is they just found it. Oh, this is an interesting bottle. And then they figured out that it had some cool stuff inside. And they with the help of some appropriate people in Australia, were actually to do an archival search in Germany and found the log of the ship that tossed it over.
Cool.
Okay, and found an entry for June 12, 1886, made by the captain, recording the throwing over this drift bottle. And you can even match the handwriting in the log to the handwriting on the message in the bottle. Okay, which I just love. So out of these thousands of bottles that were thrown overboard, apparently, this is the 663rd that was found, and is by 25 some odd years the oldest confirmed. So there you go. I wish the captain were still around so that he could
that's
know
so cool.
that his experiment worked.
So I recommend rigorously that when you open this website and you look at it, you put on in the background the music by Sting. Message in a bottle.
So how old did you say it was? 132. So what year would that be put in the water?
It was put in the water in 1886.
Oh, I think
June 12th, 1886.
I want them to look for viruses.
Oh.
Because we don't have a lot of viruses from 1800. We don't have any from 1800s. You know, the first influenza isolates were 1930. We have other viruses human viruses in 1908. But I would like to see, because it might be something on the paper. Would be cool if they could.
People probably got their patties all over it now. It's seriously contaminated.
Yeah, looks like they're touching it. It's too bad. Too bad. That's very cool. I remember as a kid, I wrote a message, put it in a bottle, and I threw it in. I said, maybe somebody will find this someday.
What did you write?
I don't remember. I think I put a cork in it, so it's not going to last, right?
I know what I would have written. I would have had a scotch bottle. It would have been empty, and I would have said, refill and resend.
Yeah. You like scotch?
I do.
Nice. Dickson, what do you have for us?
Well, I mean, I decided to make a virus pick this time because I usually don't do that. But I ran across this website from the BBC on the new World News report that had an excellent set of graphics and simple charts and easy to follow, good science communication. The whole thing was just laid out beautifully. And I think the material was as accurate, of course, as the Chinese would allow it, because they were the ones that supplied all the statistics.
It's actually well done.
Very good.
But I liked it a lot.
Nice chart,
and it re-emphasizes the Tufte approach to displaying quantitative information, basically. Mm-hmm. And this is a very good example of that, I think.
Well, it's simple, nice graphics, but it's not hyped.
It's not
right.
hyped.
Yes. And it also addresses all the questions that people ask.
Exactly.
You know?
Exactly.
Very nice.
Here's the one spread to the UK, how face masks work. But the best part is a picture of the two dogs with face masks. It is just so cute.
Precious. Did you get the email that I sent you about the artists that had a series of masks for the avoiding? And I didn't want to put it up because I thought they were making light of a very serious situation, but still, they have an unusual take on life.
So my pick is a video cast of a meeting I went to a couple of weeks ago of the NSABB, the National Science Advisory Board for Biosecurity. This is the board. It's a government board that will look at experiments you propose in case they have gain-of-function experiments proposed. Mm-hmm. And if you'd like to see how these meetings go, who's on it, what they say, I'm there, and I talk about certain issues and so forth, but mostly it's in case you're curious. And this committee will now be reviewing experiments on new coronavirus, I'm sure. So check it out if you want to see. So we had a couple of listener picks from Sophia. We have one from Stephen. It is a podcast called This Podcast Will Kill You it's by two grad students studying disease ecology, Aaron and Erin. Have the same name, huh? And so that's cool. That's a very popular podcast, by the way.
Yeah, it looks good. And it's very nicely produced. At least the site is.
And from Neva in Buda, Texas. Dear TWiVissimos, coronavirus brings China's surveillance state out of the shadows. This is a Reuters article, and it's just chilling. Listen to this.
It is.
When the man from Hangzhou returned home from a business trip, the local police got in touch. They had tracked his license plate, and from a town where they have a lot of corona cases, they said stay home for 2 weeks. At 12 days, he was bored. He went out early. This time, not only did they contact him, so did his boss. He'd been spotted near Hangzhou's West Lake by a camera with facial recognition technology. He says, I was a bit shocked by the ability and efficiency of the mass surveillance network. They can basically trace our movements anywhere. And it goes
Yeah,
on.
it's amazing. It goes
brother.
on.
This is Big Brother.
This is Big Brother in spades.
Neva says, what a world we are creating. Yeah. Also, voice to text made coronavirus corona violence. Remain calm and keep on TWiVing. Neva from Buddha.
Beautiful.
Is it Buddha? Sorry.
Right.
Yeah.
Buddha. That's right, Buddha.
Sorry about that. Thanks for your picks, folks. You can find us on any podcast player. The show notes are at microbe.tv/TWiV. The show notes have all the links to the articles we talk about and the full text of all the email we read. We cut a lot out, and we still didn't get through them all. So they're all there. Microbe.tv/TWiV. If you want to send us questions and comments, TWiV@microbe.tv. As this outbreak subsides, don't go away. Just keep listening.
Yeah, exactly.
Keep subscribed. It won't hurt.
Stay tuned. There will be another outbreak of something.
You can learn the fundamentals. And
right.
That way, next time you'll be better prepared.
That's right. There's never not going to be another outbreak. There will always be another outbreak.
For sure. And, of course, if you really like what we do, consider supporting us financially. Microbe.tv/contribute. We have ways you could either give a small amount of money a month or a large amount if you'd like. It's
all
fine.
at once if you like.
Or you can give us a bolus. It helps pay our expenses because we all do this because we love science and we love to communicate. Would that be a fair statement for all of us?
You betcha. Yep, you bet.
Dickson Despommier can be found at Racaniello.org and parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Go get your grandkid.
I saw your old colleague at the NIH this week, Bert Flanagan.
Oh, hi, Bert. Great. Cool.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.